Okay, start. Okay. Well, dear colleagues, dear students, ladies and gentlemen, good morning, everyone. I am Paola Saccomandi. I am Associate Professor at the Department of Mechanical Engineering of Politecnico di Milano. I'm very happy today to welcome you all to our workshop, Technological Innovations for the Precision Medicine of Tomorrow. First of all, let me thank Martina Delandro for the support and the hard work she did for the organization of this event. This event has been financially supported by Institut Français Italia and the French Embassy of Italy. This institute aims at fostering the scientific collaboration between Italian and French institutions. For this reason, last year, Martina applied for a call, Cassini Junior, in order to organize an event which allowed to invite speakers from French institutions in the framework of the topic we choose. Considering the scientific collaborations and partnerships in present and past projects that we have, my group has the chance to have strong connection with French institutions, and most of them are clustered in a very nice region of France, Alsace, and particularly in Strasbourg. I strongly invite you to visit this place if you can. It's a great place. So I will introduce the speakers afterwards, but first of all, I would like to mention these institutions. So the Inria Grandes with the Mimesis team, IRCAD France, and Institute of Image-Guided Surgery of Strasbourg. As far as it concerns the counterparts, so the Italian counterpart, I'm very happy to welcome today my colleague from Politecnico di Milano, from Istituto di Ricerche Farmacologiche Mario Negri, our neighbor, and Istituto Italiano di Tecnologia. As you might see, we opened the borders to Switzerland because a colleague of us who used to be in France is recently moved to Switzerland in the University of Bern. So there is more than just Italy and France today. Well, let's go to the core of the workshop of today, focus on the technological innovation for precision medicine of the future. We decide to investigate this topic because we believe that the technology, the innovation, and the progress which concern this important field of medicine deserves to be strongly investigated, and also because somehow the research of all the groups invited today is around this topic. It's, of course, a very huge topic, and if we consider, so first of all, what is precision medicine? Is the aim of precision medicine is to develop approaches and methodologies to target, to develop therapies which are somehow targeted and developed in order to consider the individual variability of a patient or a subgroup of patients. If we go back and consider the strong progresses of the science in general and medicine more in specific of the last decades, we see that we can observe a very huge and strong progress, which is based mostly on the evolution of the technology. And nowadays, medicine, if we talk about medicine 4.0, which is one of the last revolution, together with the new era, the new version of the Industrial Revolution, is based on the fact that the treatment of the patient is more and more dependent on the technology that you use to treat the specific patient. The field is very huge. That's why Martina and I decided to organize the event of today like a sort of journey, a journey tackling some specific topics. And we have decided to start from the operating theater, talking about the technology that is nowadays supporting the work of the surgeon. Let's consider the image-guided surgery, the use of smart surgical instruments which allow the surgeon to augment his or her sense in order to collect more and more information about what is going on during the operation. What is the need of that? The need is to support the decision-making process. Along with the technologies, we will also mention the importance and the role of the real-time medical simulation in this, together with the advent and the prominent role of the algorithms based on artificial intelligence, which allow to have a better prediction on the outcome of the treatment. As you may have noticed, all these points take into account the fact that there are augmented information, so there are data. How those data are produced? Well, physiological parameters that can be measured on the patient, on the operating room, in the instruments, allow to improve, to augment the information that the surgeon can use. That's why the use of specific system for monitoring these parameters is quite important. Okay, so till now we have touched many topics, and I would say that the main keyword of all this topic is represented by data, information. So what we will also see at the end of this day is which is the workflow of this data. Data for health is among the most critical part of the new revolution of medicine in this day, and it will have an important role in the future years. The afternoon is dedicated to one of the hottest topics of the precision medicine, indeed nanomedicine and nanotechnology. So we will have information inside about the cutting-edge research on nanoscale materials, use of biocompatible nanoparticles, and the functionalization of polymers for targeted drug delivery. This journey is possible, has been made possible by my colleagues. I'm very happy to welcome you today. I will introduce them considering the steps of our journey. So we will start with Dr. Michele Diana, Scientific Director of the Research Institute Against Cancer and Digestive System in IRCAD in Strasbourg. Welcome, Michele, and thank you. Then we will move on with Stephane Cotin, Dr. Stephane Cotin, Research Director at Inria, a leader of the Mimesis team in Strasbourg as well. We will have then the talk of Martina, followed by the talk of Dr. Eric Felli, postdoctoral researcher at the University of Bern, Department of Biomedical Research and Pathology in Switzerland. Then we will go for lunch. In the afternoon, we will start the nanomedicine section with Professor Paolo Decuzzi, Director of the Laboratory of Nanotechnology for Precision Medicine, Istituto Italiano di Tecnologia, Genoa. We will welcome Professor Filippo Rossi, Department of Chemistry, Materials and Chemical Engineering, Politecnico di Milano. And the responsibility of closing this session is in the hands of Dr. Paolo Bigini, our dear colleague and neighbor, the head of Nanobiology Unit, Department of Molecular Biochemistry and Pharmacology, Istituto di Ricerche Farmacologiche Mario Negri in Milano. About the program, a very brief description. So we will have 3 main blocks, 2 in the morning, divided by a coffee break around 11 a.m. Then we have an open discussion about the topics, the main topics discussed in the morning around 12. We have a lunch break, and at 2 p.m. we will start again with the single section dedicated to nanomedicine until 4 p.m. with the open discussion and the final greetings. Let me thank again the sponsors, Institut Français Italia, French Embassy in Italy, the project partners, so European Research Council and Fondazione Cariplo, and I would like also to mention that this event has received technical sponsorship from the IEEE Sensor Council Italy chapter. All right, so well, I will disappear now and I will leave the floor to my colleagues. I hope you will enjoy, and what I really wish is that this event will be a food for thought for all of you, for the speakers, so that they can meet each other, but I've seen they already done. They established a good connection beyond science, and also for all the attendees. Thank you very much. I leave the floor to Dr. Diana now. I allow you to, yeah.
So I start with the classic 2 questions since 1 year and a half. Can you hear me? Can you see my slides?
Yes.
Okay, yeah, the typical questions when doing a Zoom meeting. So yeah, first of all, thanks a lot, Paolo, for the kind introduction and for your summary. Yeah, I also would like to say that your career is absolutely brilliant, and I'm so happy for that, because you really deserve it all. So yeah, I would like to start by providing you what the, can you see the slides?
Yes.
Because there is, I don't see them advancing.
No, we see the first one, but not.
Okay.
Oh, here
Yeah.
it is. Yeah.
So yeah, I would like to share with you the vision we have at IRCAD about the cybernetically augmented surgeon. As Paolo was saying, of course, we consider that the use of technology, computer science, robotic science, can enable the surgeon to augment the senses that are required to perform a surgical procedure. So, of course, the brain to create the theranostic algorithms of diagnosis and treatment. Of course, the eyes, because the surgeon must see, and of course the end, which ultimately is the effector. I don't know what's going on. Can you see the video moving? Oh, I'm sorry, there's some problem with the connection maybe. So yeah, I wanted to also show you, Paolo was mentioning the Institute of Image-Guided Surgery, which was a project of IRCAD that we launched 10 years ago. And ultimately, in 2016, there was the opening of this brand new operating room, which is characterized by, it's a smart operating room, which is equipped with imaging technologies right in the same environment. So there is a CT scan that can jump in and out, is mounted over rails. There is an MRI system in the same room, and there is also a system that's called is a C-arm, a robotized C-arm, that can enable to do real-time imaging and also to fuse previous information gathered with the other imaging technology systems. And one of our priorities when conceiving this new operating room, this new surgeon, was also to improve the level of safety in the operating room, taking inspiration from the aviation world. And one of the projects that we launched is called Condor, that stands for Connected Optimized Network and Data on the operating room. So, of course, data. And in the Condor project, one of the aim is to obtain a synchronization of all the data information that can come from the medical and surgical devices in the operating room, including the physiology data that come from the anesthesiology machine that can be interconnected, for example, with the laparoscopic instruments. When doing minimal invasive surgery, we insufflate the abdomen to create a space, and this can generate changes in the pressure. So the ventilation, the anesthesiology side should communicate with the laparoscopic insufflator in order to, for example, correct in real time to prevent that there is an over pressure at the abdominal side that would prevent the lungs to expand. So this is done by humans so far, but if there is a way to optimize and to automatize this is really important to prevent complications. So basically, all the parameters should be controlled. That's the aim. And in the same project, the Condor project, a very nice tool has been developed. It's called Indexity. This is an annotation tool. Annotation is basically at this stage of the human-machine interface is how we can discuss, let's say, how we can reason with the machine, providing what is so far, let's say, the ground truth. So we know what we are looking into a diagnostic imaging, what we are looking into a surgical video. So far, we are at the stage of teaching the machine. Probably is a kind of human arrogance. Maybe the machine is able to do better than us, but that's what the state of the art today. And this is a great tool that is facilitating this process of annotating images, and also, as Paolo was also mentioning, to understand the workflow. This is extremely important as well. So this Indexity tool can, for example, you can annotate the presence of a surgeon, the moment the surgeon jumps in. So basically, you can cure all the videos that are going on during a procedure, and gently building this atlas that enables to teach the system, the algorithms to let's say understand what is going on. And for example, this is a nice application, very recent publication from the group of Nicolas Padois at the IQ. They use this data to, for example, detect the surgical phase. It's a very preliminary and very basic, maybe, outcome so far, but this is extremely important. This is one step, is a building block to start this process of human-machine interaction in the surgical field. So they used hundreds of videos. At a certain point, the algorithms were able to dissect the surgical procedure, which is here a basic one, is a cholecystectomy, in several phases, and this was very accurate in detecting the surgical phase. That's been published in the top surgical journal, which is Annals of Surgery, just this year. And this has a lot of applications as well, because it can enable to optimize the resources. For example, it can send a signal to the world saying the case is about to get finished, so get ready for the next patient. It can generate autonomous reporting of the surgical procedure, which is something that's quite time consuming for a surgeon to dictate the report right after the procedure. So it's a building block, but it has already some application, some concrete application. And the other interesting point on which we are working is, of course, to the so-called radiomics, so the use of CNN, machine learning, deep learning algorithms to improve the diagnosis, to automatize the diagnosis of a pathology. And also, this is really something that IRCAD is working on, the IRCAD team is working on since years, is the modeling. That Stephane will also discuss about it. What I can say is that from the team, recently there has been a spin-off created. It's Visible Patient, which produces those beautiful models of the entire human anatomy. And the very important thing has been that during last year, during the COVID, by the way, and all this process of improvement started to improve the diagnosis of COVID. So they were working on CT scans of the lung. They could optimize so nicely the ability to do the reconstruction that they moved from the 4 hours that was the time required before up to 9 minutes nowadays. That's a great improvement that has been achieved. And these models can be applied during the surgical procedure to obtain the augmented reality, so the virtual image that's fused with a real patient. That's a nice case that was done at the Institute of Image-Guided Surgery. And as you can see, the Professor Mariano Jimenez here is a percutaneous surgeon. He's a surgeon that does interventional radiology procedures. Of course, this is a sort of surgery. If you can burn completely a tumor using a sort of energy, that's surgery. And as you can see, there is a fusion of images, so you can navigate into this image with a ultrasound and have those 3D reconstruction, which is really beautiful and helpful to guide the gesture. And of course, the augmented end is all about robotics. And we are partnering with a lot of surgical robotic companies. What we think is very important is to integrate the images into the surgical robotic system. Here you have an example of optical imaging, so fluorescence image-guided surgery, and the integration of those models as a GPS that the surgeon can manipulate to guide the procedure in real time. Because robotic assistance is not really producing the improvement of outcomes, probably because we are only using the telemanipulator to replicate what we already can do in minimally invasive surgery. So to really extract the maximum, to really optimize the use of the robotic system, first we need to implement imaging technologies and probably cognition. And so moving towards probably the next frontier of robotics, which is automation. And we are working a lot on this. Also, again, as you can see, there is a flow going towards this, the implementation of artificial intelligence algorithm, this instrument reasoning, the implementation of biomechanical modeling, all to really make in a way that the machine can sense what's going on. And we have launched very recently, the surgical data science team of IRCAD, a huge project that's funded almost 5 million funds to do, it's called Disruptor, to enable a percutaneous injection of a needle to perform biopsies or to energy-based ablations, fully robotized. And the interesting part of this project is the fact that it's a really heart-grounded using echography, sonography, ultrasonography, which is a real-time imaging modality, widespread, and cheap, relatively cheap. So this is something that we really think will democratize the percutaneous surgical approaches. And I move now to, that's quite futuristic, and I move now to what is my topic of interest since 11 years. 11 years and 3 weeks, because the topic is precision. When I first had the in my hands a prototype of a near-infrared imaging system. So I'm working mostly on surgical optomics, so the use of optical imaging during the surgical procedure. And we created a team that gathers together the chemists, the hardware people that do those devices that produce light into the near-infrared wavelength that penetrates deeper into the tissues, so enables to see things that are deep-seated, and the surgeon can visualize them. And of course, more recently, we need this software augmentation, the use of deep learning, machine learning algorithms is all producing nice progresses into this field. So what is optical imaging? We can distinguish the exogenous fluorescence. So those near-infrared cameras, they detect the signal that comes from a substance that's called fluorophore, and that can be administered to the patient. So a fluorophore is a substance that, upon illumination with a specific wavelength, will emit a photon into a different wavelength. That this signal is the one that is captured and displayed to the surgeon. And the fluorophore can be as specific as the most commonly used, it's the indocyanine green, or can be targeted. It can be a monoclonal antibody that goes specifically targeting the cancer tissue and display so augment the ability of diagnosing the cancer tissue, so to enable a radical removal of the cancer, for example. That's exogenous. And there is a lot of systems, so the vast majority of companies producing imaging systems for surgery have moved into this field. So that's the list. Is a very recent review that has been done by one of my previous PhD students. So basically speaking, there is the entire manufacturer list that's now moving into this field. And so with as said, with this fluorescence imaging, you can improve the diagnosis because you will see better the tumor tissue, for example. You can prevent complications because this enables you to see structure, critical structure that the surgeon should not injure during the surgical procedure. It can improve the radical removal of cancer. You will see the entire cancer tissue and you will be able to remove it. That can improve the staging. Let's say a bit more surgical, too surgical probably. It means to see the extent of the tumor in real time during a procedure. And for example, to prevent complication, we are partnering with some drug companies like Lycor that produces a dye that can go into the ureters. The indocyanine green is only excreted by the bile, and this is excreted by the kidneys as well. And you can see this beautiful peristalsis of the ureter, so it will be, let's say, much more difficult to injure it. It's always possible, but if you can see it so nicely, probably you will spare it. And together with Andrei Klimchenko, my associate, is a chemist, leader of the bioimaging group, we have produced a polymer that is embedded with a fluorescent molecule that is 30 times more fluorescent than indocyanine green. And we are using this as a painting to coat surgical devices. So we wanted to escape the regulatory, let's say, have a faster path towards clinical application by coating a device. So this is not a drug, it's not administered to the patient. It only covers the devices. And for example, we have used in a lot of applications. We have already published many times, but one very nice is, for example, this, to coat the ureteral catheter. And so you can see we tested it in a porcine model and also in a human cadaver model. This gives you this very bright fluorescence that will protect the ureter from surgical injuries. And another very nice topic of the use of fluorescence is to assess the perfusion of the tissues, because a non-perfused organ will not heal. So doing a resection of the bowel, you need to assess the perfusion. And this is very difficult to do by naked eyes. And when you inject this fluorophore, you will see where the perfusion ends. And we have created a software that is even capable of quantifying the amount of fluorescence and gives you a so-called angiography effect, so you can store the image. And this has been, in patients, this has been predictive of 5 complications. All could have been prevented, but this is not yet a medical device, so the surgeon could not use this information. We were only observing what was going on. You see, after injection, the software computes the speed at which the fluorophore has reached the bowel, and the faster, the better. So that's a little bit how we can summarize it. And you see, we were overlaying this analysis onto the real-time images. And with these boxes, we are assessing where the surgeon should make the resection. I repeat, the surgeon could not use this information. This was only for us to see how accurate it was. And you see, that's the resection lines are displayed. And the surgeon was placing a clip where it was intending to do the resection. And we were also placing a clip where the software was intending to do the resection. And actually what happened was that the software was, as we were, that was the hypothesis coming from our preclinical studies, that the software was more accurate. And look at this case, this is very nice. So you see the clips were placed by the surgeon where he was about to make the resection. And then we injected the indocyanine green, we let the software run, and the software was suggesting to resect 2 centimeters more proximal. And actually, the nice part of the study was that we were getting samples of capillary lactates, which is a marker of perfusion. And you see that there was 2 centimeters difference between the software intended resection and the surgeon intended resection. And actually, the lactates, so this parameter that tells you if there is perfusion or not, was 6.3, so it was much higher in the human intended resection, and 0.5, so extremely well perfused in the software intended resection. And so we were talking about exogenous fluorescence, but there is another very important part. It's the endogenous fluorescence. There are many technologies that can do that. Is based on light tissue interaction. For example, here is an example of hyperspectral imaging, which is a device for now available mostly in open form that sends light to the tissues, and the tissue, the light undergoes different scattering, diffusion, reflection, and all those interactions can be used to quantify some compound of the tissue, like the hemoglobin, the fat, the water, and all this is a kind of a chemical analysis of the tissues and can provide a perfusion evaluation, but also a signature, for example, of tumors or many other things. So yeah, the open device we're using in the OR, but also very recently we tested the minimal invasive version, and we have been using it in humans, of course, to test the perfusion of colon, and it's really accurate. We also the system so far displays the images only side by side, so we used augmented reality techniques to overlay this augmented information onto the real time, so to co-localize the augmented information provided by hyperspectral imaging onto real-time imaging. This is a software that's called Hyper, so hyperspectral-based enhanced reality. Paolo is smiling because she knows it. And we compared fluorescence imaging and hyperspectral imaging. Actually, we found that the algorithms of prediction of lactate in the experimental model was more accurate in the hyperspectral. So we are really moving more towards endogenous fluorescence. And in the clinical setting, we have launched this trial Ex Machina. We have 70 patients so far included, and this is a autonomous recognition of tissues. And I will show you some examples. You see, here is the spectral signature of a tumor of the colon. The colon has been open because we don't have yet an endoscopic system, so it has been open. You see there is a real great difference in the spectral signature between the tumor tissue and the healthy tissue. And with 10 cases running several architecture of CNN, of a convolutional neural network, we achieved an accuracy of 93% in discriminating the tumor and the healthy tissue with only 10 cases. So we expect really that this accuracy will be enhanced by accumulating more cases. And look at this. This is all the same data set, but we annotated some cases of liver tumor, and we did the same thing. We ran several algorithms, and also the unpublished data is a sneaky preview, and we achieved sensitivity that could be improved, but a very, very high specificity of 95% in discriminating the healthy from the tumor tissue. And that's also another study that we are running. It's called PREVENT, that stands for Precise Recognition with Enhanced Vision of Endocrine Neck Targets. We aim at recognizing the nerve and the parathyroid glands. We have 4 glands that are situated across the around the thyroid that should not be injured. And we are doing by hyperspectral imaging. You see, we can really nicely identify the parathyroid because it has a different composition. It's made with there is colloid inside, so it has a different signature. And the aim of this study is to acquire an atlas of spectral signature of all the endocrine targets. And that was very nice. So we are doing by autofluorescence. You see the parathyroid gland as glows spontaneously when illuminated at 820 nanometers without any contrast injection. We achieved a 98% visualization rate. And by hyperspectral, we did this new concept that's called CNN-based augmented reality, in which the parathyroid gland could be visualized in 92% of overall accuracy. Again, it's only 14 cases, so we expect that this can be improved. And by augmenting the exigence, let's say, of the model, the parathyroid gland can be visualized as a single dot. And yeah, the nerve, we were not good in visualizing the nerve, but actually in one case, yes, that's displayed by the forceps, and that's the hyperspectral signal, and that's how it can be visualized. There are some limitations as more structure. There are a lot of confounders that are the same that we have by our own vision. But we were optimistic because we were coming from an animal study in which we used larger nerves, the vagal nerve, and the accuracy was pretty good. That's is a preprint stage. It's not yet published, but you can read it. And yeah, so we think that with time we will be also able to see the nerve, which is a crucial target to recognize and to preserve during neck surgery. Last thing, this is, by the way, this is the thing that fascinates me the most, is the use of monoclonal antibodies that reach the cancer tissue specifically. And we are now partnering in a big consortium. It's called the CAST. We are using the anti-CMET, which is expressed specifically in colon cancer. And this illuminates so brightly and so nicely the cancer tissue. And we, in the CAST project, which is a Marie Curie fellowship in the Horizon H 2020, the aim of the CAST is to avoid the surgery if there has been a response of the tumor upon radiochemotherapy. That's all, and I thank you for your attention.
Thank you. Thank you very much, Michele. Amazing, as always. Can you switch? Yeah, okay, thank you. Is there any question for Michele from the floor? Please raise your hand and we'll give you the right. Yes? Paolo,
Perhaps
please.
Paolo, I'm sorry for the coughing. Very briefly, because I know that it will be a dedicated session for Q&A at the very end, but it was really super interesting, Michele. I'm looking forward to perhaps have a chat later in the next few days, and perhaps also with Paula about what you have been presenting. So my understanding is that the endogenous, let's say the endogenous signal is associated with the specific features of the tissue, and therefore, when we look at cancer, the increased cellularity density of the cells, the increased amount of collagen, and the more dense network of extracellular fibers would actually be captured by the camera, by the spectrum, basically, and it doesn't really need at this point the introduction of an exogenous fluorophore. Is that? I guess that's where you are going, right? Or the fluorophore will be always needed to a certain extent to confirm the data that the hyperspectrum is giving you.
This is a phenomenal question, and it goes really to the point. So basically, we still think that there is a complementarity in using both. So in assessing with the endogenous system, also an exogenous signal. We have worked on this topic. We have even developed what we call the correction algorithm, because when you inject a fluorophore, the endogenous signal is completely wasted. I don't say wasted. It's completely corrupted, and we found a way to come back to the situation before the injection of the fluorophore, because the fluorophore will certainly have a specific wavelength in which it becomes excited and a specific wavelength in which it will emit. And in this band, from the excitation to the emission, the endogenous signal is corrupted. And but we think that there is something to extract even from this corruption of the signal. Playing in a clever way is like using still it is a contrast, and so probably there is a sense in using both. But this is really, we are first comparing both endogenous versus exogenous with targeted fluorophores. That's an initial aim. This is in the cost that we are running. It's a huge consortium, it's 24 partners, and then after having compared, we could come to the question you were asking. Is it usable or not? We think that there is still some use, probably to see better the margins, but this is a open question.
Great, thank you. So then, as you said, we have a dedicated session at the end of the morning, and I hope, the discussion will go on this sense. So thank you, Michele, for now. See you later. Thank you very much. And now I would like to leave the floor to Dr. Stephane Cotin with this talk on the role of the mathematical modeling for a patient-specific treatment. Welcome, Stephane, and thank you for being here.
Thank you. I will try something. So, buongiorno a tutti, sono lieto di potervi presentare il nostro lavoro. Grazie ancora a Martina e Paola per l'invito. Hope that was not too bad. So I will talk about mathematics and numerical simulation.
Stephane, sorry to interrupt you.
Yeah.
We see maybe the second screen, or we don't see the full in the presentation your presentation modality.
So you don't see the main screen or whatever?
We see, the first slide and the other slides on the side.
Okay, I will stop the sharing and start again. Let me know. That works better.
The same.
So it's not full screen. That's what you're telling me, right?
Screen,
yeah. Okay. Usually it works. What can I do? I turn off my camera just to see if it makes any difference. Stop the sharing. Start again.
It's the same. By the way, for me, we can go on with this as you like. We see very well. It's not screen, but we can see very well.
I'm just afraid that when I have videos, they won't play if I'm not in a full screen mode. Okay. Let me try once more. Wait a minute. Still the same?
Yes.
Man. Okay. So I can make that bigger, maybe?
Yes.
I don't know if it makes any difference. You tell me.
It's fine. It's fine.
It is fine? Okay. Let me continue then. Yeah, for the videos, we'll see how I can handle that. So again, thank you for the invitation, and I will switch to quite a different topic compared to what Michele just presented, but I think somehow this is maybe somewhat connected thing, and it's about more numerical simulation and mathematics, and how basically models can still be used at the time where data plays a more and more important role in medicine and in the tools we can provide clinicians. So just a little warning that there will be some images about surgery, but I think after what we've just seen, you've already been warned. So here's a kind of layout of the different things we do in the team. And again, it's not necessarily the only representative of what we do in my group, but it's just the idea that using numerical simulation, we can provide different ways of assessing what is going on during a surgery. We can also train, guide clinicians, and even provide control to robots. And the underlying part in all of this, at least for us, is the idea that there is a model describing, well, a phenomena that we can model. And because in many of these applications, we have interactivity and we need real-time feedback, there's the question of how we can provide real-time simulation with those models. So it leads naturally to, I think, a very trendy word these days, which is the idea of a digital twin. And I will just give you various definitions, and I think at least among those, there are important ideas that to me really make sense when we talk about digital twins. The first one is it's a bridge that we can create between the physical and the digital worlds, okay? And so by digital twin and model, we can create a replica in a way of many things. It can be an organ, it can be a process, it can be an object in other fields such as, I don't know, car industry. But that this connection between the real world and the model is really the key of these digital twins, in the sense that from sensors or imaging of the real world, we can improve the digital twin, and this model in turn can provide added feedback to, in our case, the clinician. And this loop that we can build in this case is kind of a virtuous cycle. And I think that's where we can do lots of very interesting things. So in our case, again, if we focus in the field of medicine, we want to use and develop digital twins to train surgeons or other clinicians, to plan complex interventions, to guide during these interventions by providing feedback, visual feedback often, but we can also control medical robots or even design new medical devices. And in many of those cases, the requirements that we are looking for, so the main characteristics in a way we want for those digital twins is that they are accurate, they are sometimes real-time, and usually when we have this constraint of computation time, we want that they are both accurate and fast. And I think the key thing again is that those models bring a predictive capability, so we can go beyond what is available in the data. We can also see in the future, such like you know, when you look at the weather channel, you want to know the weather of tomorrow, not of today. And so when we look at that, we take a either narrower window, because it's a very large field, and we look at computational anatomy, and in particular biomechanics, but also a little bit of physiology. And we looked at models of the eye, the liver, the brain. And we've looked at phenomena such as electrophysiology and bioheat transfer, and I will give you examples of that. So in here, there are videos that unfortunately will probably not play, but it's okay. They are just there to describe different topics we worked on in a more visual way. And again, in all those cases, at least nearly all of them, we created numerical models of different anatomical structures, and we designed, developed numerical tools so that they can be computed in real time, and you could also have interactions with them, okay? And maybe the next slides will show you a little bit more of that. So if we look at the biomechanics of a tissue, there are lots of different things that can be investigated. It usually starts by building experimental setups, so we can create curves like what you see on the right-hand side. And these curves, they basically describe the relationship between the stress and the strain. So how much force you need to apply to tissue to generate a certain range of deformation. What these curves show here is that this is most often non-linear, and also that depending on each patient, and sometimes also on the pathology, you get different responses. So what this tells us is that the models are complicated because they are non-linear, and I provide here an equation not just to show off, but to say that, you know, these are not simple equations to solve. They naturally, nearly all the time, lead to partial differential equations that need to be solved. And there is no analytical solution for those, so we need to develop numerical techniques. But they also depend on parameters, such like mu1 and mu2 in this case, that have to be identified to really characterize a particular patient or behavior. And so if we try to get a solution for those equations on a particular shape, domain, that's what we call omega here for the liver, then we have to solve numerically these equations. And for that, we very often use the finite element method, which is well grounded in, I would say, mathematics. So we know that the smaller elements we use to discretize the initial shape, and the more accurate is the numerical approximation of the continuous solution. But in turns, it leads to a very large system of equations that we need to solve. And this system of equation, in the case of a nonlinear problem, which is, again, most often the case, will depend on the solution. So we basically have to create a very large matrix, which changes every time step. And we have to inverse a system involving a very large matrix. And that under a set of boundary conditions that describe basically what are the constraints around this organ, or in the problem in general. And so we developed lots of methods, or not lots, but we developed methods where we try to find the best possible trade-off between the accuracy of the solution, so basically how finely we discretize the model, for instance, what model we choose to represent a particular problem, and also what kind of algorithm we can provide, and on what kind of architecture, using, for instance, the GPU, to be able to speed up computations that are usually, in the order of a minute or more, and we want to reach computation times that are less than 50 milliseconds, and sometimes even less, okay? And so this is one big area on which we have been working on, and we keep working, and I will show you some results on that. And then if we want to create these digital twins, we also need to learn how to generate in the most possible automatic way that numerical model, not just the shape of the organ reconstructed from the preoperative data, but we need basically this tool chain to be as automated as possible. So basically from the CT scan of a patient, we could, or MRI, we could have a numerical model, so a digital twin. So here are some examples that basically cover the different levels of complexity maybe we can imagine for a digital twin. The first one is in the field of training. So in surgical training, we need models that describe the actual behavior of an organ, but models can be generic in the sense that they represent an average anatomy, or they can represent different pathological cases, but they are not patient-specific, okay? And we looked at that on many different application levels, but the maybe most interesting one is the case of the eye, which appears to be more complicated than we initially thought. It's made of, the eye is lots of different membranes, in a way, interacting with each other. They are sliding, they are nonlinear, they are, under pressure, and when we interact with the surgical instruments, we introduce lots of modifications in this. So we developed here a combination of modeling choices that were able to reproduce the phenomena we were interested in too. So we made some hypotheses and simplifications that were, we thought, good choices for the application we were looking at. And also we developed numerical methods to be able to compute these very quickly. And well, I think I can maybe play this like that. So here are some, I don't know if you see the video, but it's some results on interactions with these different numerical models using a haptic device. So it's a robotized system that can allow us to control the instrument, but also allow the clinician to feel, or the trainee to feel the forces that are applied or computed by the simulation, okay? And here, hopefully you get to see this one. There is sound as well. I hope it works. Do you have sound? No?
No.
Okay, well, you can read the subtitles, but it's just, you know, a little summary of a project we did a while ago on this eye surgery simulation. So you see on the left-hand side the hardware that was used to visualize the virtual world, to interact with it, and to feel back the forces in a very accurate way. And this is done in a very ambitious project with a foundation based in the US, which is called Help Me See. So basically what this is just explaining is how this system can be used by clinicians to learn this technique, which is a new surgical technique aimed at being developed, deployed in developing countries. And we changed the training, usual training approach, where rather than using cadavers, animals, we provide a completely virtual environment on which every trainee will be able to do many, many training sessions up to a certain point where we can assess the skills of the trainee, and then the trainee will go on a set of 15, 20 actual operations on a patient under supervision before being, you know, able to actually do procedures on another patient. And so in this case, the project is finished. The system has been deployed in different training centers, and validation studies are basically coming back saying people actually learn something from using a system like this, which was the intended goal, but you know, we wanted to prove it, and the first results are coming back very positive. So now, if we have a way of modeling complex anatomy like the one of the eye, and we can create a simulation which is very fast, you can interact with it. The next step is to go towards this idea of a digital twin, is to make this simulation patient specific, okay? And this is a topic we have worked on for a while now, and here's an example of an application, but I will show you others. Let's say we want to provide an augmented view of the anatomy of an organ like the liver during a surgery. So we want to show, while the liver is being manipulated, where the internal structures of this organ are located. So we need to track the motion of the surface of the organ using computer vision techniques, and then we want to drive the numerical model from this information and show the internal structures. So we need a model of the organ. We need first its geometry, and we know that today using deep learning techniques or other existing tools, it's getting more and more easy to generate a three-dimensional representation of the organ. So this is something that has made lots of progress, and we build upon this. What we are providing now is ways of, as automatically as possible, generating a representation of the physics, the biomechanics of this organ by integrating information from this pre-operative data, CT scan, MRI, to create a numerical model. So not just a shape, but something that can behave sort of quote unquote like the real organ, okay? And If that video works, you can see that at this stage, it is possible to, even on a tablet, load the anatomy of a particular patient, visualize it, but also interact with it and start planning the surgery or understand, its behavior or how the liver will look like at the time of the procedure and better understand how to prepare for the surgery. But we're not yet at the stage where we have a real patient-specific model, because to do that, as you've seen before, we need to integrate in the equation parameters of the model that are specific to the patient, so that basically the deformation based on the forces we apply is really specific to that patient. And this is a work that we have done very recently, where we use Bayesian methods. So basically, it's kind of machine learning, but I would say old school, but it works really well. We're using videos, live videos of the surgery, so basically images acquired at the beginning of surgery, and tracking some motion of particular points on its surface. So here we facilitate the job of the algorithm by placing some markers on the surface of the liver, and then we track them. So you see those blue points moving, at least I hope you see them moving a little bit. Those points have their counterpart, they are green in the middle, and the virtual model of the liver. And the model, without the right parameters, will provide an incorrect result. At least there will be a difference between what the model predicts and what we observe in the image. And so using a predictor-corrector strategy, so some kind of Kalman filter, nonlinear Kalman filter, we will adapt the parameters of the model so that the model prediction matches as accurately as possible what we observe on the real organ. You can see here that, for instance, this curve is describing how the initial stiffness of the liver, which was set too low, can be corrected using observations from the real world. And this is really the kind of things that really describe what a digital twin is and the benefit of combining models with data. Here, we use a model to estimate a stiffness which was not measured through observation. So the model provides this link, exactly like it was described in the definition, between parameters and observations, and that opens up lots of different things. So these are old videos of work that we did in the field of laparoscopic surgery, where in this case we don't parameterize the model yet, but we provide this augmentation of the liver, so we see what is inside, but using patient-specific models. So this is a reconstruction of the anatomy, and then we fit this model and we track the motion in the images in the video. It's two-dimensional or stereoscopic image, and using information just about the surface of the organ, we provide information about what is going on inside the organ. There's another video behind this image, but let me see if I can, yes, I can show it this way. This is another example, but in the field of open surgery and using different, another way of capturing data, okay? And again, it's the same idea. Key information here is that we provide, again, an enhanced information to the clinician using this combination of data and model. And we can do this in other, I would say, surgical applications. So just a few images to show you also what we've done in the field of neurosurgery here. What we want to do is show where tumor is located during a neurosurgery by using information from the superficial vessels. So we first need to identify the vessels that are just appearing underneath the surface of the brain. And then here we combine this information with a model which is hidden behind this green box. And the model basically provides the deformation of the local region of the brain such that knowing the new position of the surface vascular structures, we can compute an updated position of the tumor inside the brain. And you can see that it was initially thought to be where you have the blue outline, and the model predicted it was where you have the pink outline, and it was the right thing. So if we leave for a minute the world of biomechanics, we can also combine models and data in the field of medicine by looking at other phenomena. And the one I wanted to just talk about for a couple of minutes is heat diffusion. Okay, and the first time we looked at that was in the context of cryoablation. So the problem here was to understand how heat generated, or it's in this case cold, generated by needles that are placed inside an organ, like in this case it was the kidney, how these needles, depending on where they are located, how many we place, would generate a sufficiently cold region to be able to destroy the tumor. And so what we did is we created an interactive tool. We're using preoperative data of the anatomy. We could create a three-dimensional model of the kidney and the tumor. We could then place interactively the needles you see on the right-hand side, and using very fast GPU-based finite element models to solve the equation you see above, we could predict the different isosurfaces describing the regions that would be behind the threshold we are looking for in terms of temperature and other regions. So you could interactively change the location of the virtual needle to understand how many needles and how they should be placed to create the best possible cold region near the tumor, and then try to replicate that in the actual procedure. Problem here, or the limitation I should say, is that the model was not using parameters specific to that patient. So we could predict something very quickly at the time of the procedure, but it was missing the patient-specific aspect. So this is something we have worked on in the context of the Laser Optimal project with Paola. And here, the problem is slightly different, but the methodology is similar. Here we want to destroy tumor using a laser, and the question is, what is the temperature in the neighborhood of the laser impact point, and how could we create a model that tells us what is the heat map using very sparse data. And this sparse data is provided by small needles that are placed near the laser, and we are limited to the number of needles, so 2 or 3, and these needles, they use FBGs or FBG arrays to be able to measure the temperature. So what we've developed here is a model similar to the previous one, but this model will learn from the data, even though we have very limited data, to improve itself. And you can see here in this curve that so the temperature, the real temperature distribution is the green curve. Okay, that's what we experimentally measure. The model, without being parameterized from the data, predicted the black curve, which is quite different from the green one. And then using data assimilation, so basically by learning from the data and feeding back this information into the model, but by basically improving the model, we got the red curve, which is quite close to the green one. And you can see here at the bottom that the error, prediction error, is quite low. And so basically, the maximum error is below 1.5 degrees. So with a model like that, we can provide, we believe, meaningful insight to the clinician. And it's really a digital twin in the sense that the model replicates in the best possible way the physics of the phenomena, but it's not perfect. And so it's learning from the data to get better, but as it gets better, it gets also more predictive. And that's really a key feature we are looking for. So to sort of conclude, what we believe are some possible future steps, well, at least the ones we are looking at or even working on, are related to, well, this idea that, we have data. It's not perfect, but it's there, that we collect at the time of the procedure most often. And we need to be very fast in our computation to solve all those different problems you've seen. And so we came up with the idea of using deep learning in a very different way from what people usually do, which is not to analyze images to provide, I would say, higher level interpretation of these images, but to actually train a neural network to predict the solution of a complex computation like the one of a finite element method. And I don't have time to go into the details of this, but it works. And here I can show you two videos. They should be playing side by side, but of a liver deforming under some constraints. And it's on the left, the result of an actual computation. And on the right is the result of a prediction from a neural network. What's very interesting is that the prediction from the neural network is 500 times faster than the FEM computation, meaning that we can start using more advanced and more complex models that would otherwise be way too slow to be used in the different contexts we are looking for. But the other very interesting thing is being a neural network, this is a natural model to inject data, okay? So in the same spirit as what I showed before, our next step is to take this solution provided by a neural network, which is, for now, I would say generic, and use data collected in the operating room to make the model more predictive and more patient-specific. And this is what we are starting to work on at the end of the summer. So to finish, I just want to say that I strongly believe that it's really important to combine data with models, and that these ideas of digital twins are really key in medicine. But it's clear that human biomechanics and physiology are very complex. Every patient, every person is different. So we need, on one end, better models to better interpret and learn. And we need to find ways of better exploiting medical data. But for instance, what Michele showed previously clearly highlights also the fact that there are so many ways of collecting information in the operating room that we need to also look into that and bridge the two things together. I clearly think that data alone can provide lots of insights, but by combining this data with models, we can, basically increase the power of the data. And just a super quick word on the fact that lots of the things we do in the team are made public or can be made available in any case upon a request in a very large open source code base that is called SOFA. So this is something we are developing. We have been developing for the past 10 to 12 years. if you're into computational modeling, simulation, and the kind of things you've seen, this is a good toolbox to play with. And if you're interested, don't hesitate to ask me. And I will stop here and just thank you again for the invitation and remind you that we are organizing MICAI, which is a very large conference on the kind of topics you've seen this morning. It will be virtually in Strasbourg. It's very unfortunate that we cannot have it in real, but at least it's a very good forum for the kind of topics I think this workshop is about. Thank you again.
Thank you, Stephane. Thank you very much. It was very interesting. Any question? We have a very short time for a question from the floor. If not, okay, so I will make one, but we will discuss maybe more in detail afterwards. So can you give us just one example of the way patient-specific parameters influence the prediction of a model? Can you give a quantitative example in terms of accuracy and how much the ground truth is different from the prediction?
Well, I can take again the example of the liver deformation, but any kind of, I would say, biomechanical model. So in the kind of application we look at, where you are in the operating room and the only thing that we can see is usually the surface of the organ. What we want is to predict what is going on inside the organ. Okay, so for instance, what is the motion of a tumor during a surgery, during a therapy? And the motion of the tumor inside the organ relates to what we can observe on the surface based on those parameters that we are talking about. So it depends on the equation, the model we use, but it depends on the parameters we put in that model. And I don't have like an exact number in mind, but I would say that with the wrong parameterization, we can get probably errors up to 1-2 centimeters, where with the right parameterization, we can probably get below the 5 mm accuracy that most clinicians are looking for. What we saw in the project we work on with you in the temperature assessment is a bit similar, right? So clearly, injecting data in the model is a way of reducing these errors. And but again, not only it reduces the error, but it makes the model able to predict the evolution, the time evolution of a phenomena in a more accurate way. So I think that's really the thing I wanted to convey to you guys, is that if we had data but maybe no model, maybe from that data we could get an idea of, okay, what is going on right now, okay? But if I want to know what will happen in a second, in a minute, it's the same with, again, the weather predictions. I mean, it certainly got much better with more data. But if you don't have a model, then you cannot really predict things in the future. And I think this idea of predicting is really key in medicine.
Yeah, I agree. Thank you, Stephane. I see there is a question from the floor. May I ask you, Mohammed, to make your question in the open session?
Thank you, Dr. Stephane, first for your interesting lecture or presentation.
Mohammed, can I ask you to make the question afterwards?
Yes.
Because we are running a bit late on the program.
Okay.
So we have a dedicated session for this.
No problem, no problem.
I will be there, no problem.
Yeah,
Thank you.
Stephane will be there, so thanks. So thank you, Michele, thank you, Stephane. Now it's time for a short break. We will be back in 20 minutes, so 11:20 with the second part of the morning. Thank you. See you in a while. Okay. We will start in a while. Can you hear me well?
Yes.
Yes, great. We had a lot of problems today. I would not say anything.
Yeah, I don't know what happened with the full screen thing, but I hope no one has this issue anymore.
All right, so we will start with the second part of the morning, workshop of the morning, and I will invite now Martina Delandro. She's a senior PhD student in my team, and she will give us a talk about some measurement approaches for real-time intraoperative control. So the floor is yours.
Good morning. Good morning, everyone. I will share my screen, try to share my screen, hoping that everything will work. Okay. Working? Yes, I think so. Can you hear me?
Yes.
Because okay, I will because we cannot check if you can hear, but in case, just stop me.
Wait 1 second. Switch off your microphone and.
It's working. Okay. Okay, so today I will talk about innovative measurement approaches for a specific real-time intraoperative control. Before starting, actually, just a few words about me. In particular, I'm a PhD student at Politecnico di Milano, and during these years, I had the chance to collaborate with French institutions. In particular, I spent a few months in Strasbourg, ITU, and in ITU Lyrique in Bordeaux. And during this time, I could benefit from working in multidisciplinary environment. In particular, this was a great opportunity for me and my professional growth. And this is why, with Professor Saccomandi and in collaboration with the French Embassy, we organized this workshop, in particular to promote new collaboration and exchanging between students and also workers and, of course, researchers coming from different backgrounds, in particular from Italian and French institutions. Okay, and I will now move to my main project, for my main talk. First of all, again, thank you everyone for participating to this workshop. Thank you to the speakers who are today with us. In particular, which is my project. I'm working in a European Research Council funded project, Laser Optimal, whose main goal is to achieve an accurate tumor removal by minimizing the damage for surrounding healthy tissue. In this panorama, my PhD goal is to develop innovative approach for real-time intraoperative control or laser ablation thermal therapy. To better understand what I'm talking about, let's make a step back. In particular, intraoperative control. What? Do you have some problem?
Yeah. Can you? Yeah. Sorry, I ricondividi.
I will try to show. Laura? Okay. I was saying that my main PhD goal is to develop an innovative approach for real-time intraoperative control of laser ablation therapy. In the specific, what does the intraoperative control means? In particular, it's an approach to control in the specific an operation, in surgical operation. When and why we should introduce this terminology? In particular, the advent of intraoperative control is directly linked to the advent of minimal invasive intervention. Minimal invasive surgery refers to any surgical procedure performed through tiny incision instead of larger opening. Compared to traditional surgery, we have seen several examples in the previous presentation. Of course, during traditional surgery, the surgeon has a direct feedback, a visual feedback during the operation itself. In case of minimal invasive surgery, the surgeon has to use a tool, rigid tool, to reach the target and operate. Which are the benefits of this minimal invasive intervention? These are cost effective. In particular, we have reduction for bleeding, reduction impact on patient, reduced hospital stay, and also most of the time this can be the only option for non-surgical candidates. Which is the main drawbacks for this minimal invasive surgery intervention? Again, in this case, lack of feedback for direct feedback for surgeon, and so the imaging navigational assistant technological solution, as the one the previous speakers shown, are crucial for orientation, guidance, and control. Today, in particular, in my talk, I will focus on thermal therapies as minimal invasive thermal procedures. In the context of interventional oncology, thermal therapy refers to application of thermal energy for the purpose of focal tissue destruction, and in specific for management of local tumors. We talk about focal tumor destruction. They are mainly classified into laser ablation, microwave ablation, radiofrequency ablation, high-intensity focus ultrasound, and cryoablation. The common feature of this therapy is the induction of tissue increase to induce necrosis of the cells. In particular, the working principles are different. For the laser ablation, in the laser ablation case, the basic principle is the conversion of laser light into heat. When interaction between laser light and tissue occurs, we can have partially light reflected and partially light can penetrate the tissue, and under this light undergoes mainly two phenomena: absorption and scattering. These two phenomena are the one responsible for tissue temperature increase, then for, in particular, heat generation. This application of heat causes cells necrosis. In particular, the heat generated in the tissue and the consequent damage for the cells at membrane and subcellular levels, actually it's influenced by several factors. In particular, here I just reported laser characteristic can affect, of course, this heat generation. In particular, wavelength of the laser or laser settings, or the emission characteristic of the applicator can affect the heat distribution in the tissue, but also tissue characteristics, so perfusion of the tissue, as well as optical, thermal, and electrical characteristic of the tissue itself. All these factors are, of course, affecting the prediction. This means that predicting this heat generating, this heat distribution in the tissue is very hard, it's very tough, and this is why we need to control in real time, which is the actual effect on the tissue during the treatment itself. When we talk about real-time control, this should be specific, I mean patient-specific, organ-specific, but also area-specific, so according to where we are actually ablating. How we can achieve this specific real-time control? Measuring, which is the effect we are really inducing on the tissue, measuring the temperature and the thermal damage which we are really applying on the tissue. Before moving to, in particular, before showing you which are the innovative technology in this this field, I will show which are the main strategy for perform laser ablation. In particular, one possible strategy is the percutaneous approach, where the inner organ, the tissue, is reached through via needle puncture of the skin. And in this case, for inserting this needle, CT ultrasound can be used as imaging guidance. In the other case, another approach provides the use of an endoscope, in particular when we want to reach, for example, pancreas as target, so pancreatic tumors, as well as left liver or caudine lobe of the liver. This can be the right approach to perform. In particular, in case of endoscope, we mainly refer on ultrasound endoscope approaches, where the approach blends the use of endoscope to visualize inside the gastrointestinal tract using the lighting in a specific camera, and also ultrasound probe to better have an idea of which are the organ structure nearby. Another approach, but of course in this case we don't talk about minimal invasive techniques, is the open surgery one. Sometimes the ablation therapies can be performed during open surgery. Which are the main steps of the procedure? In particular, if we want to perform a laser ablation, nowadays we have as first step the planning step. So the patient is in particular undergoes CT, MRI imaging, and during this step the tumor size, quality, and count is performed. In particular, for example, the surgeon can then define the strategy to implement how many applicator can be used. For example, if the tumor is larger than 2 centimeters, the surgeon can define to use 2 applicators in parallel position. Then the targeting step. So once the strategy is defined, the applicator are placed under a CT ultrasound guidance mainly, but also MRI can be an approach used. And of course, monitoring, as I was just mentioning before, is essential to control what is happening during a laser ablation treatment. In this regard, for this step, there are several strategies, in particular imaging-based. In this case, we distinguish between a qualitative approach and a quantitative approach. I will explain better in the next slides what I'm in particular what I mean with these quantitative and qualitative approaches. And also temperature sensors and optical approach are innovative techniques possible. Then the last step is the imaging examination. So which is the final real effect we could induce on the patient. In particular, in this case again, we could use imaging approach with CT or MRI imaging, and in this case also what is important is that sometimes we can have inflammatory reaction, and we should wait for time to really understand which was the effect of the therapy. This means, in particular, wait for 45 to 70 days after the treatment to understand how much it was really effective. Okay. Okay, then which are the traditionally techniques used nowadays for the implementation of laser ablation technique? In particular, if we talk about lung, bone, and abdomen, strategy can be to use computer tomography imaging, where we have an excellent visualization of applicator using fast high resolution and axial imaging, but also in this case, computer tomography images suffers from low contrast for lesions in soft tissue, and also from ionizing radiation. For the treatment of soft tissue, the approach used nowadays is ultrasound imaging. In particular, this is a not ionizing, flexible, and cost-effective tool, and in the use of this ultrasound imaging, in particular, for the monitoring step, we mainly refer on contrast enhanced ultrasound, where the lesion is visually defined, in particular, thanks to contrast relationship between the healthy surrounding tissue and lesion, but this is just a qualitative control of what is the real effect we are inducing, and can suffer from poor contrast and also microbubble formation during heating, as well as limited ultrasound propagation across interfaces. This is why we need more quantitative and reliable technique for really measuring what is happening in the tissue during our treatment. The most promising tool in these regards is the MRI thermometry. In particular, there are several parameters which are sensitive to temperature in the MRI. I just reported here an example based on proton resonance frequency shift. In particular, this is the most used approach because it allows for linear temperature dependency and also not tissue type specificity. Starting from the phase images, in particular of the MRI system, it's possible to measuring the shift of this phase image. We have the current phase, we have the reference phase measured before the heating, and then from this shift it's possible to extract the temperature map. Which are the limitations in this case? We can just obtain a relative temperature information, not absolute one, and also proton resonance frequency shift is prone to susceptibility errors. Which is the main ability of MRI technique, the one to image in any scan orientation with good spatial and temporal resolution. As I reported here, we were able to reconstruct the temperature for the 3 orientation, thus allowing the volumetric reconstruction of the temperature effect, the tissue temperature increase. And also for all the pixels in the image, it's possible to reconstruct the temperature curve. Of course, this is not enough. Starting from these temperature images, we need to obtain the thermal dose, the finer thermal dose, the thermal damage really inducing on the tissue. And for this, models based on Arrhenius are used in the clinical practice nowadays, in particular in the neurosurgery field. I just here will show you an example of MRI thermometry monitoring during an ex vivo laser ablation treatment, where in particular we have this overlay of magnitude image showing the anatomy of the tissue and temperature maps. Mainly here, this is the main slice showing in particular the higher temperature because it's closer to the laser applicator. And in this way, we can reconstruct again the entire volume of interest. So just to summarize, magnetic resonance imaging is the great opportunity, holds the great opportunity to perform laser ablation treatment in one single integrated environment. All the steps could be potentially performed inside an MRI machine. In particular, for the laser ablation, this is very sweet. It suits because MRI suits because of compatibility with the laser applicator. Also, in particular, it is not ionizing machine and allows for volumetric temperature maps. Also, of course, it allows personalized and precise measurements and also, it's possible to have high soft tissue contrast as well as functional and metabolic information for precise targeting. Which are the limitations in using MRI machine? we just can have indirect measurements of thermal effect. Also, currently, the efficacy of such therapy, of such treatment, of such technique has not been yet demonstrated to date for any thermal therapy. So much more trials are needed in this regard. And also, MRI machine is high cost and present a lot of problematic related to artifacts in the images due to motion and also need for ferromagnetic tools. As last part of in the last part of my talk, I would also show you some other innovative techniques for intraoperative control of laser ablation treatment. In particular, here there is an example involving the use of temperature sensors. Already Stephane mentioned fiber Bragg sensor. With my team, we are actually investigating the use of these sensors for this purpose, and which is the main limitation. In this case, of course, they are invasive. To obtain temperature information, they should be in contact with the measurement site, but however, we can say that they are characterized by attractive features. They are biocompatible. We talk about small dimensions, around 200 micrometers as the diameter, and also the ability for multiplexing. It means that we could have a lot of sensor in one single fiber optic. And here I just reported an example of our use of these sensors in vivo case. We inserted a fiber optic embedding 8 sensors in parallel position respect to the laser applicator, and we could monitor the temperature during the treatment. Here, a second example, in particular in an ex vivo case, where in particular the temperature was reconstructed using 12 optical sensors, embedding 420 sensors. So we could reconstruct it in the volume, the temperature increase. As last point, I would mention just briefly another approach, a very innovative approach for intraoperative control of laser ablation treatment. This is based on optical technology. In particular, we are investigating the use of hyperspectral camera. In particular, Dr. Michele Diana were mentioning this tool. We also try to use this camera for intraoperative control of laser ablation. This camera holds the ability to collect spectral and spatial information. In our first preliminary analysis, we checked and we saw that the spectrum of a tissue can vary during a laser ablation by increasing temperature, and this variation is actually localized in a specific spectral band associated to chromophore of the tissue. If we just focus our attention on methemoglobin chromophore, here I reported images collected in the range 600, 700 nanometer, so the one associated to methemoglobin during the laser ablation procedure, in particular during temperature increase, and it is easy to notice that this methemoglobin content is varying during the procedure, and in particular this variation is also depending on the position of where we are really considering our pixel. If we are in the boundary or if we are in the center, the behavior of this content during the laser is changing. This is also visible in a 3D graph where we clearly see that the methemoglobin content at the beginning decrease in the area of ablation, but after 70 Celsius degrees it starts increasing specifically in the center of the ablated area. And we see actually this lobe increasing in the center. So we investigated the use of this specific methemoglobin or other actually chromophore as indicator of the thermal effect we are inducing during the ablation. Just to sum up, today I briefly talk about thermal ablation as minimally invasive thermal procedures. We have seen that accurate tumor removal requires real-time specific control. Nowadays, the imaging techniques are based on just qualitative lesion identification as control strategies, but more quantitative real-time control is necessary based on temperature and thermal domain measurement. I talked about MRI thermometry as most promising approach in this regard, but also other approaches are under investigation based on temperature sensor or optical tissue response. Promising results encourage, of course, further study in order to identify which is the best approach possible. And of course, this best approach is patient, organ, and area specific. Thank you.
Thank you, Martina.
I will stop some sharing. Okay.
Great. Thank you very much.
You can.
It was very interesting. So we have time for one short question.
I have a short question. Do you hear me? Yes. Okay. I think I understood, but I wanted to double check what you said about the MR thermometry in the. So because at some point you said relative measurements, and then you said indirect measurements. So basically, you don't get absolute values of the temperature, right?
Yeah. We cannot get the absolute temperature. We just have increased relative information. When I mentioned the indirect, I was talking about the effect. I mean, we can measure temperature, but we use a model for predicting with the real thermal damage induced. In these terms, is an indirect measurement of thermal damage induced, but it's of course direct measuring of temperature as a delta. It's shift as a relative increase.
So if you were to place, like, if you had at the same time one FBG sensor to measure an absolute temperature, and then you have the MRI thermometry, could you then, calibrate your heat map somehow?
Sorry. Yes, actually, what is done currently is use other kind of fiber optic sensor, fluoro optic sensors, placed, of course, close to the area of ablation, and to actually then get the absolute increase of temperature. Yes,
okay.
It's perfectly true.
Thank you.
Thank you.
Thank you. Thank you. Thank you, Martina. Now I would like to invite our next speaker, Dr. Felli. He will give us very interesting insights about the flow of the data according to European regulation.
Thank you very much. Can you hear me? Oh, no, if you can hear me? Yes? Okay. Now, next challenge. I need to see if I'm able to show the PowerPoint. Can you see the presentation? Should be there. One second. Can you see it on Teams? Okay. So maybe I can just see. Paola, if I give you the presentation on the PDF, I say next, and so we solve the problem. One second. But this is the old one, sorry. You copied. No, this is the old one. So I'll give you the new one. Okay, so I can start. Okay. Dear all, thank you very much for being here, and thank you, Paolo and Martina, for inviting me here at this event. My name is Eric Felli, and I would like to make a very brief introduction on the barriers of European health system in advance of advent of precision medicine. Next. Until now, most medical treatments have been designed for the average patient. As a result of this one-size-fits-all approach, treatments can be very successful for some patients, but not for others. Precision medicine, on the other hand, is an innovative approach, as we know, that takes into account individual differences in people's genes, environments, lifestyle, and so on. This means that precision medicine deals with the problem. Sorry. This means that precision medicine deals with the problem of no responders, or with those people who have an adverse response. In order to obtain such a specific treatment, the whole patient's population needs to be divided in clusters that share similar clinical profile. In this way, it is possible to treat people depending on how they on the disease expressed, considering their genetic profile. This clustering action has some deep ethical and political, social, and economic consequences. Next. For instance, the EU policy in general are facing with problems related with the clinical world, such as the need of a new medical regulation or the need of the national system. From a medical point of view, as the diversity of treatment increases, health insurance systems are challenged by precision medicine. Indeed, the ability to discriminate patients due to their genetic profile is directly linked with our ability to predict who is the most probable type of profile that will develop a certain disease, and then who is going to be more expensive for the system, either public or either private. Until now, no regulation for predictive treatment is available. And this leads us to another problem, which is the universal access. Precision medicine is still expensive, and applying it as it would mean to filter only rich people or member states in some areas of the world in general. Moreover, patients' rights are not clear. As precision medicine advances, many personal data are generated, related, hosted, or databased, retrieved, raising a problem of data security and privacy. Finally, the complex of such change is important. Indeed, many different policies in many different fields needs to be coordinated and linked all together in order to obtain a fair and shared system without creating a discrimination based on clinical profile. Next. Considering the problem related with data, the digital transformation of health and care in the digital single market identified three main priorities. First is the easy and secure access to their health data across the borders, of course, the need of shared European data infrastructure, this is really important, and the need of a better empowered citizen with digital tools. I would like to draw your attention on the shared European data infrastructure, because while the ethical point may be solved by a common shared and harmonized policy, which is still challenging, I have to say. In EU, we need to consider that national barriers and diversity among member states in terms of health systems are really, really strong. But in general, a new infrastructure for this would implies to have a common health system generating a European patient, a European workforce, and a European health data infrastructure over which it is possible to generate a private-public partnership under the framework given by the central state, which in this case should be the EU. Next. Another public consultation made by the EU showed how citizens asked for immediate application of this innovative tool and to develop a disease prevention action to have international standards for which only integrated system could be the answer. to have an unbiased and public health orientation. So, I mean, it seems that they really want this technology now in their hospital, but they want a fair treatment for everybody, you know, this is a fingerprint of European culture. Precision medicine is much more just an innovative tool. It stimulates many changes and involves many stakeholders. It's easy to see how from precision medicine, a revolution in a clinical field, we are already in the revolution of the whole European system and the national borders. Precision medicine involves deep changes in our political life and create many opportunities to improve patient health and for a new internal market. Next. Nevertheless, as you can see from the last report of the EU Parliament, there are much more basic problems to solve. For instance, we still need to understand the risk of precision medicine that precision medicine could bring. Additionally, the continuous problems raised by the national health system of the member states are challenging the advent of precision medicine in Europe. Next. So then, in 2018, I tried to highlight these problems in the report that I had the pleasure to coordinate in Brussels in the European Parliament. Next. The idea was to furnish to the European Commission an integrated map, or at least a draft of the map, to understand how complex was the interaction between artificial intelligence, robotics, precision medicine, and of course, the logical consequences like the reduction of hospitalizations and costs. Now, we know that European health systems are under stress, especially during pandemic. COVID-19 just showed us what we knew. An aging population, changing the societal behavior, and the rise of chronic disease increases the long-term cost of healthcare. Together with the budgetary constraints, this puts immense pressure to the stability and sustainability of our healthcare system that cannot be solved merely by throwing more money them and a single nation or in every nation. Or if you put more money in Italy, you put money in France, you know, in Germany, you will have just more expenditure because the problem is not solved in a systemic way. So fortunately, digital health technology can improve our health service. Advances in robotics mean so all these types of new technology mean a benefit from improved health outcome and actually they provide for a high quality of life. Health systems see a reduction of healthcare costs, improve the quality of service and overall effectiveness. Indeed, as ex-Commissioner Andriukaitis, with which we had the pleasure an honor to collaborate, recently noted, digital tools can be used to improve health in Europe through promotion, prevention, and protection. However, the success of digital transformation of health depends on how we embrace this change in the next years. Next. Access to the quality of affordable healthcare for all citizens should be the ultimate goal for each and every society. We have to consider that everything is happening very fast. The European Commission adopted its first e-health action plan in 2004, followed by a second action plan in 2012. Several initiatives and legislative texts have supported these plans, including the famous 2011 Cross-Border Healthcare Directive, the Commission Green Paper on mobile health, and Commission staff working document on telemedicine. While the Estonian presidency of Council launched the Digital Health Society in July 2017, which eventually led adoption of Council conclusion on Health Digital Society. So then the European Parliament has also adopted the same strategy and a resolution of civil law rules on robotics among the many. Momentum for digital health in Europe is clearly building, but for the EU to take a leading position in the future health technologies, we need to take action now. So despite proof of benefits that digital health technologies offer, structural barriers hinder their availability to patients. The lack of infrastructure, and this is really important, and long-term planning are two barriers that need to be break down in the next year. On the other hand, proper infrastructure can make health digital. On the other hand, a long-term vision will rally around our partners, public, private, and to make our health system fit to the future. At that time, we have highlighted the need for structural changes, proposing the European Public Health Area, a brand new multi-annual financial framework, which included single market, digital and innovation. and the complete revision of GDPR. I was really happy when I saw that finally the EU decided to properly fund this field and support these changes. Without the money, a policy is only a concept, as we know. Next. We also founded the day dedicated on health system in Brussels with the participation of European Commission that tried to analyze the healthcare cross-border directory, which coordinates the right to patient to be treated in every country of EU, regardless where they came from. So the need to fight for a patient in Germany, let's say, to have the access to Italian healthcare system returns us how far we are from a European public health system and how far we are from having an integrated precision medicine system approach to more monitoring in the hospital. This is because the data, as we discussed it before, needs to be centralized in a unique infrastructure. So this is a process that is still ongoing. Regardless the many advances made by the European Commission and the member states so far, still uncertainties surround patients' rights. Thus, through the continuous effort from the Commission, the Member States, under the rules of the European Court of Justice, cross-border healthcare is a process that still needs to be debated, fully integrated, and fully needs to be integrated in the directive in the healthcare sector tissue. The European health dimension represents a collective need to become more and more necessary, considering the social and economic challenges that nowadays the EU is called to run. The EU healthcare nowadays is a collection of many national healthcare systems, each of them with a profound and rich history, with many different excellent aspects. This makes you one of the richest powers worldwide, culturally speaking, in this sector. Besides the welfare concept that is at the base and shared among the many different European cultures, increases the potential of a possible unique system can be a new way that is an unprecedented. It's not just like USA, because we don't share that culture. Okay, our culture is just better, and I'm not saying just because it's in terms of be superior or inferior. It's just because our idea of innovation is innovation that is delivered to everybody, regardless their social status. And this is something that is a value, is not a tool. There is a big difference. So for us, it's a polar star. It's not something that we need to reach. It's a starting point. Next. The world is digitally transformed, and the health industry is leading this process. The EU, it means us at the end, must grasp this opportunity to become the world's leading continent in digital health by ensuring the modern and future technologies are available, affordable and acceptable. Adoption of these technologies will not only benefit the health of Europeans and guarantee the sustainability of our health system, but furthermore make Europe attractive for industries, create jobs, and establish the European Union as the leader in digital health innovation. So then finally, for integrated and fair precision medicine based on a health system, we need the European public health infrastructure data, European public health area, and European public health workforce, which means a completed European Union. And I want to just add just this. Why we need to run, and not just because the world is running, that it would be like a reason for this. It's because there is also a macroeconomic feature that is important to understand. BCE cannot keep buying our economy. We are supported by the Draghi's plan, and in this moment, the cost of the money in terms of loans and so on from the banks is really low. So this period needs to be used by everybody to review the industrial field, to invest in technology, in small, medium enterprises, and so on. When this period will finish, the cost of the money in general will be much higher. And this means that it will be harder for everybody to review the whole industrial part. So we can speculate scientifically how much we can. We can show the nice video of super augmented reality, which I did. So I'm a big fan of that. But if we are not able to transfer this to the clinical side as a product, and immediately it's just offering know-how to the rest of nations that are more wise, they are just going faster than us. Indeed, the last cover of The Economist just show how they make these nice pictures, you know. And he said that there was this big car that were just over the European, and one of the two car one is USA and then in China. Why? Because they invested in precision medicine, in artificial intelligence, in robotics as anybody. Not just in the development, not just in science. They just investing to transform this in an end product. Next. Just thank you very much. I don't remember because I don't have the presentation with me now. Okay.
Thank you very much, Eric. Thank you very much. Yeah, it was a very nice point of view from yeah, your vision, no, your vision, the situation from the point of view of structure, yeah, the problems that and the challenges that are still present on this. Well, okay, so thank you. We are perfectly on time. So I will open now the sort of the virtual roundtable, and I will start by inviting back Michele and Stephane, Martin and Eric, yes. And we had a question before I had to stop from Mohamed Aref, so can you ask your question right now if you want?
Yeah, yes, Dr. Paula. I was asking Dr. Stephane about one of his slides inside his interesting about the brain when he was diagnostic the brain tumor. Then the mathematical model was recorrecting the place for its identification.
Yes, I see which one you are talking about. Maybe, yes.
Yeah. Could I ask about certain part in it?
Yes,
how you know about the actual place? Like you were saying that it was in certain place, then you recorrected with the mathematical model. Yes. And this was in real time, as I think?
It was. I will just bring up the slide so everybody sort of remembers this part. Do you see it? Do you see anything?
Not this one. I think it was one slide.
It was this one, right?
Yeah, this one.
But so I need this one. Well, I need both to explain. But so what happens is during this kind of surgery, there is this phenomena called the brain shift, which is due to the fact that the brain inside the skull is under some kind of a pressure because you have the CSF, which is between the brain and the skull as a damping mechanism in case of a shock. And when you open the skull, when you do a craniotomy, this CSF leaks out, and it results as a shift, a deformation from the brain, okay? And so this brain shift is known to have an impact on the location of the tumor. So basically, the preoperative imaging showing the location of the tumor is no longer exact when you start the surgery because of this brain shift, okay? And the shift could be, a few millimeters, and it could be much larger. It depends on many parameters that we don't clearly understand. And so what we did is we use the information about the vessels that are on the surface of the brain, at least the ones that are closest to the surface of the brain, and we can image. And we create a biomechanical model of the local vessels, I would say. And that model relates the location. So basically, you can see the local vessels as being an anchor to the location of the tumor. So we know that they are going to move together. And so if at the time of the surgery, we see that the vessels have shifted, then we can compute the new position of the tumor. And so what we show here is that that motion was significant, that we can recover it. And the question you had maybe was, okay, how do we get the first outline? Well, the first outline is basically by saying, okay, the skull, which is rigid, at the time of the surgery, we can use it to register the preoperative image. Okay, so this is what we call the rigid registration. So that's usually what is done. Okay, so we use the shape of the skull to reposition the preoperative data onto the patient. But since the brain is not rigid and is shifting because of the opening of the skull, we see that the result of this classical registration here, the outline in blue, is no longer correct. And so we correct it by using the model. And so you have a much better idea of where the tumor is located. So this is the story behind this.
Okay, but the tumor is more related for the pathological tissue reporting, because when you're working with the tissue and it's interactive with the imaging, you have several interactions due to the light interaction with the tissue itself. So even if you're working with the veins or the artery, which is like supplying the blood for these tissues, it will be distracting a little bit, because you're going for further bit for the, it's called what, supplying blood for these cells, and it may be more active or highly active, and this moving forward with the imaging with the phosphorus, which was in another session, which if you like injecting it with certain dye, and you could see it. But here what I see, you take the arteries or the vein like in demigration to could be find the actual place. So the surgery is working, and when he is working and removing the tissues, after this you see it with the ground truth with the pathological report or something more. Like here, after you extracting, because this is the brain and you open the skull as you were saying, so you're taking like an initial image, is this right? And after this they working, and after they finish you take another image and you like comparing them with your mathematical model.
Exactly right. Yes, okay. It's MR imaging that we take in both cases. And just to be clear about that, this is a work done with a group in Boston, the Surgical Planning Lab. So I have a former PhD student who is doing his postdoc there. So this is a joint work with them. But I'm pretty sure that the I would say validation is performed through interventional MRI because they use it quite a lot. But it's cumbersome. It's a little bit what also Martina was saying, you know, working in a MR scanner is not so easy. So they would like to avoid using the MR scanner at the time of the surgery. So that's why we are looking at it. You said something which is clearly true, which is that for now, as you can have guessed from my presentation, we are really more into the mathematical modeling, the biomechanics, the numerical methods. We are not into imaging. And we know that we should probably start looking at other imaging modalities like multispectral imaging, fluorescent imaging, but we're not experts in this. So certainly if some of you guys want to discuss about this with us, I think there are probably ways of improving this kind of approach by using better images, showing more information. The more information we'll capture from the intraoperative imaging, certainly the better the result.
Yes, indeed. Could I ask another question about another system you was talking about it?
If Paula allows it, yes.
Yes.
About the eye interaction system, is this a commercial system which is for training?
It is in a way a commercial system, but in a way only because this foundation that I was mentioning called Help Me See is basically for now not commercializing the system. So so we created the research, we created a proof of concept, then we started up a company 8 years ago. Now this company has finished the simulator, which underwent a series of, I would say, validation studies. And now this foundation is basically building simulators to deploy them in India and in China, but they are not commercializing it. It's a foundation which is basically raising money to manufacture the simulators and basically make sure that through that we can train lots of new clinicians in developing countries to treat cataract. The whole purpose behind this is that, because I skipped many slides in this, is that cataract blindness is basically touching 20 million people in the world. Okay, so there are 20 million people in developing countries who are blind because they do not have the ability to have the same surgery we have, you know, in France or Italy, in Europe, in the US. And so the whole project is about promoting a new type of surgery which can be easily deployed in these countries. But we need to train about 30,000 people, almost like technicians, not even surgeons, to be able to do this intervention. So this simulator, many of them will go in different training centers in China and India for now. And the idea is to train very quickly lots of people that, and then, you know, they will provide the surgery. So this foundation is covering all of this. They funded the development, they will fund the deployment and the creation of the training centers. Maybe one day it will become a commercial product, available for others. But for now, it's prioritized, I would say, for this foundation.
So it is like a non-profit foundation?
It's totally a non-profit foundation. Yeah, the guy you see in the video at the beginning is he's a billionaire, and he really wants to push this through.
Thank you.
Sure.
You know, dissipative equipment, which is from Germany, which one of the researchers was working with it in, I think, in Leipzig. I think Dr. Michele Diana, which was showing it for us. This was a research in the beginning from like 6 to 7 years ago, and now it is a commercial equipment.
I know a company called VR Magic. They are based in Germany, and they are providing a commercial product which is similar to this, but has differences. And that's why this Help Me See Foundation wanted to work with us, because their product was not exactly what they wanted. The big difference, just to finish quickly on that, is that the other existing system uses a fake eye made of silicone, and you actually put instruments in it, and they are tracking the motion of the instruments, but they are not providing haptic feedback. Okay, so you don't feel the forces. And for the guys of the Help Me See Foundation, they thought it was really important to provide a sense of touch. So this is why we have
type of simulators we have in our country. You have like a complete patient simulators and eye simulators for this part, which is maybe from silicone and other materials, which is like giving the impedance and the strain.
Oh, cool. Oh, we should talk more about that. Okay.
Thank you. Thank you. Sure. I really appreciate your help. Thank you.
Thank you.
Thank you. Thank you very much. Aref and Stephane. We have another question from this floor. Please,
Hi, Stephane. First of all, I would like to tell you that yes, when you spoke about different types of imaging, I mean, we have Michele Diana. You can ask him. Hyperspectral had very amazing results. I mean, depends on the type of depth that you need. Also, because more than 5 millimeters, you maybe don't have great results. But it's a very good tool to start with before to move in the multispectral. So you decide what is your range, and after you build up the multispectral over the range that you have selected already, the results of your experiments.
If I can answer that, anything better than just what you can see with a normal camera would already be very good, because as you probably know, at least for the liver, the surface of the liver is quite uniform. I mean, of course, there is a little bit of texture, but we know when we want to provide this augmented reality, we have to be able to track the motion of the liver as the liver moves in front of the camera. And just tracking the motion of the liver is very difficult with, I would say, classical, videos coming from a laparoscopic camera, for instance. So I think multispectral, hyperspectral imaging and other things that you or Michele are working on can bring at least this, but even the ability to see a little bit underneath the surface of the organ could also be valuable. But we've never looked into this much. And certainly, the liver being a big organ, the deeper we could image, the better. But I'm sure this could bring a lot of useful information for the computation that we do after that. So
I can anticipate you that, especially on the liver, hyperspectral imaging works very well. We made the first score with artificial intelligence of liver viability, and also we analyzed the tissue textures in different parts of the liver, and the hyperspectral imaging was very useful to discriminate the different stiffness in the tissue, only just due to the pressure of the blood. Imagine when you have the different texture, steatosis, in cirrhosis and so on. And that leads me to asking another question. In a setting in which you can have like microscopic images, is it possible to apply this model to the extracellular matrix to simulate or the different stiffness of extracellular matrix in the liver? Is this model useful for that?
No, because at the microscopic level, the model we use is more a macroscopic level, okay, model. So we characterize, I would say, the deformation of the liver, but at again, large scale. So for now, we would need other models to be able to look at a more microscopic scale, like you are mentioning, which is something we have never much looked into, but that could be maybe feasible. I don't know, what's the resolution at which you would operate?
I would say 20 micrometers. I mean, the problem is that when you have there is a big problem in the fibrosis staging. There is a lot of variability between patients, staging of the disease, and so on. And there is not any mathematical model that can help actually nowadays the discrimination, to help the pathologists to discriminate different types of stiffness and relate that with the specific staging of disease. So I was just thinking to extrapolate what you were saying.
Yeah, of course.
Other field, in a very microscopic environment, see if it was possible to exploit.
Well, I mean, I honestly don't know, but I would be more than happy to at least understand more and discuss about this to see. I know that there is someone in Strasbourg as well, working on, I would say, high resolution MR imaging to be able to relate, to compute some kind of elastography, but at a relatively small scale. I don't think it would be small enough for what you are talking about, but if you want, we could
maybe changing the tool to investigate the tissue, you know, maybe MRI is not good. Okay, thank you very much.
Sure.
Thank you again. I would like to ask something to both Michele and Stephane, because you presented your research activities and your field from two points of view. Of course, it's normal. Michele is a surgeon, Stephane is a computer scientist, and but in terms of the kind of information, so that you really need from the side of Michele and the side to make your treatment patient specific, and from the side of Stephane to make your model patient specific. At which stage we are, and what you desire for the next step? So which kind of parameters, which kind of physiological quantities you need? Maybe Michele can start.
Yes, go ahead, Michele. We need to think about that.
So, during a surgical procedure, there are several, how can I say? So to operate means, of course, to achieve the results that that has triggered the need to operate, but also to prevent complications. So, and we can distinguish two type of informations, and the one is, can I see better? So anatomic information, and so to prevent to touch structures that are not that should not be touched, that should be preserved. And the second one is more physiologic and pathologic, so what differs from the normal. And we cannot really, so both are fundamental. So you need together this information, both this information. And personally speaking, I really think that optical imaging is the tool, the number one tool. If you give me only one possibility, only one thing to use, that's what I would pick. And it's not because I got passionate about this, it's because this is really the imaging tool that fits perfectly well with the surgical workflow. It is real time, so you don't need to do a 3D imaging, reconstruct, then display. That's nice. It's fabulous to look, very sexy, but it is disruptive. It disrupts the surgical workflow. This does not happen with optical imaging. It is portable. You have the images in real time. It is made for it. And I'm in surgical innovation since almost 20 years. And what I used to say in dedicated talks with surgical audience, what I used to say is that after 1987, with the introduction of a minimal invasive approach, there have been a lot of innovations. Robotics, single site, nodes, so natural orifice, endoscopic surgery. Well, none of those, and there is a cumulative evidence, none of those has brought real impact on the surgical outcomes. But that's what I used to say, and the evidence is now coming to support what I'm saying. Surgical optical imaging is providing this. There is a consensus conference that will be done in November in Barcelona with the largest European Society for Endoscopic Surgery promoting this event, there is evidence that surgical optical imaging is reducing significantly the complications, providing this enhanced physiology information, this biological augmented reality. This is a biologic augmented reality. Is the clearest thing that we can obtain so far. Is maybe less sexy than augmented reality as Stephane is mostly working on, but that's real. It's not that augmented reality is not real, but it's augmented reality. This is reality. This is a pure biological effect that you see a fluorophore that goes with the flow and becomes fluorescent where it is. So yeah, it's providing this relevant impact on surgical outcomes, preventing anastomotic leak in colorectal, preventing anastomotic leak in upper GI. For gallbladder, so the prevention of bile duct injuries, there is a huge amount of evidence that works in terms of showing clearly the anatomy. There is not, and there will never be probably, the evidence that will prevent the injuries, because to design a randomized clinical trial to demonstrate, it will require like 20,000 patients. This has already been the case with other imaging systems with the same aim, but the axiom is very easy. If you see better, there is no, this is not rocket science, but if you see better, you probably will reduce greatly the risk of injuring. And that's what it is. But for anastomotic leak, it's clear. It's almost 60% reduction of anastomotic leak in colorectal surgery, and the publication is imminent. So, well, that's what I think. There's a level of augmentation of the surgical information thanks to optical imaging that enables to see the invisible. The surface imaging, but still invisible because it comes from deeper. I cannot say it's enough, because the more the better, but I really think that this has demonstrated to help.
Thanks, Michele. Yeah.
I don't have much to add, and you know, I'm not a surgeon, so I certainly trust everything Michele said. No, but I think that we. So I think, to answer your question, Paola, when we are at the time of the surgery, I would completely agree that the more we see, the better, or the more the surgeon can see, the better. I think that a possible role that I still see for, you know, something mixing data and modeling is before the procedure. So we can better prepare, which means then, of course, we need to control. And that's where I see today still, I would say, much room for improvement, for just one reason. Everything that Michele showed is information you can collect at the time of the procedure. And I feel that, at least again, if we look at on a routine basis, the information we collect prior to the surgery is so poor. You know, I've been told from, you know, almost the beginning I started to work on the liver, that the main imaging modality is CT scan, injected CT. And then I had a guy a couple of months ago who came to our group and gave a talk on what he's doing on MR imaging. But like MR imaging, there are so many things you can do with MR imaging. So he's using different sequences, and he's basically fusing the information provided by all those different types of sequences in MR imaging, and it's so rich. So my guess would be, let's do the same in preoperative imaging as what maybe we can do at the time of the surgery. And I think if we do this and we combine that with models, maybe we can do even better. And more importantly, I think, plan for the intervention in a way that, again, we provide this added information for the clinician. And then if we have that on top of what Michele showed, I think we'd be in a very good place. Yeah, I know there's lots of noise around me, sorry.
Okay, Stephane, you're done?
Yeah, I'm done. I stopped my mic, sorry.
Yeah, thank you. I was also...
I guess, yeah.
Go Paolo, go
No,
ahead.
Paolo. No, I was, you know, and this is probably something that can be of interest to Stephane as well as you, Martina, and perhaps Michele to a certain point. So I was really, I mean, struck and thrilled by the image of this lever that was actually undergoing bending, right? It was deforming under, you know, kind of a bending load, and you clearly show that by optimizing the algorithm and basically introducing, in addition to the governing equations of physics, also some artificial intelligence component, you were able basically to accelerate the solution of the problem, going from that was amazing. I mean, 1,000 a second, 1.5 second, all the way down to 3 milliseconds, right? So that can really become a real time in operating room tool, right? My curiosity is really these. What are the conditions on the regulatory side of the equation? And let me clarify this. I mean, this is beautiful, right? I mean, you can really have real-time information on, you know, how to proceed and how much to push on your tools and where to cut and so on. But I guess that my point is here: are we losing anything by going down from 1.5 second to 3 millisecond? And if we are losing that, is this something that can be critical in terms of I mean, it might affect a patient A more than a patient B, and how regulatory agencies are dealing with this. Perhaps my question is a little bit, you know, complex, but really we can. It boils down to what are the regulatory aspects in the OR when it comes down to combining modeling with artificial intelligence and expediting the calculation process, computing process?
It was very clear, at least clearly. No, but I think the general point you are raising is that I think we don't know exactly where we stand regarding this, because it's new, and I would say for now there is no clear regulation. And you know, what happens if a surgeon were to follow blindly what an algorithm is saying? This is where I think I see a direction that, which at least to me makes sense, is I think whether the algorithm is fast or slow, whether we spend, you know, years developing a better model, there will always be an uncertainty about, you know, the prediction made by an algorithm. So if our goal is to say, okay, you know, I want to precisely locate a 3 millimeter tumor in the liver, so I can, you know, target it with, let's say, a needle. Well, it's a very ambitious objective to make sure that for every single patient, we will always be accurate. But I think what we can do is say, okay, it is very, very likely to be here. And then that's where we switch to one of the tools that we can have access in the operating room in terms of imaging, and then to look for that. So for now, because again, we are far from being experts in the imaging side, what we, for instance, have started to do is say, okay, well, let's use an ultrasound probe to search. So if you have an intraoperative ultrasound probe and you have to search for a tumor in the entire liver, that could take a while, okay? But if I tell you, this should be around here, and you just have to scan a local area, I think as in a clinical workflow, it gets better, easier to use. So I have a thing that a way for now to go around that is to say, okay, let's pinpoint where the information we're looking for is located, if we're into navigation during surgery, and then use one of the imaging modalities that, for instance, Michele presented to then, look for the existence of that tumor or nerve or vessel in this area, in this region. Maybe this is a way to not only go along the current regulations, but also make sure that whatever machine learning or model can predict, we always have a way of making sure that what we look for is there. Just my guess.
Thank you, Stephane. Well, if there is nothing else from the floor, no question. And Paolo, if you are satisfied with the answer.
No, the reason why I'm asking is because I think Eric, right, he actually said, guys, accelerate, put together projects for launching new startup companies, because sooner or later the Draghi plan will disappear, and then we will be less competitive as other countries. And Eric clearly identified a number of limitations and things that might actually somehow slow down the developmental process. But another one is the regulatory approval. And especially when you are approaching them with something that is very cool, very new, some level of education of the regulatory agencies is really actually important. And we have seen that with nanomedicine about 10 years ago. We're still seeing it. I guess that the pandemic has helped us, right, with a couple of vaccines that are nanoparticle based, to pass a number of steps and accelerate a number of steps. But I guess it's always the problem with the development of new technologies that needs to be applied to healthcare is the final regulatory approval. So I was just curious to know what was, the level of interactions and development, also that part of the equation, which is actually very important, probably as important as the actual technology. So I'm satisfied with the answer, but it was just an open comment more than a real question.
Well, this is a very interesting point, and it's very technical. If you want to have more details, these are dynamics that are really related with the specific detailed environment. But what I can say that I share with you the same impression, and I want to add something. I think that nowadays PIs, professors, scientists, researchers are synthesizing a lot of many different types of backgrounds, and they're producing innovation just because of this. I think that the similar cultural synthesis didn't happen in that other side. So probably most of the time they really do not understand what are you presenting. That's the problem. And even though they sometimes rely on a technical, support from people that should be expert, most of the time they are expert in a specific field, but they don't have the big picture in front of them. And then they are not bad. It's just they are limited. They should no. It's like this. They should really review how they select the people that are supporting this. This is my opinion.
You said it correctly at the beginning that there are so many different solutions, and it's impossible to cover, with a few regulatory scientists, whatever is currently developed. So that's why I think on this, the scientist should keep in mind that not only needs to develop the science, but also start talking as soon as possible with the regulatory scientist and explain them the technology. That's the only way. We have seen that happen in the nanomedicine. That's the only way to proceed. And I think that the regulatory scientists, you know, they're doing incredible work. They are conservative, of course, as you said, and they need to be conservative for our benefits. But I think it's extremely important to start the education process and involve them since the very beginning. So I don't know. I mean, these are considered devices. I think they are reviewed as devices. How they are reviews, or there is a special section for the softwares. Out of curiosity, I mean, EMA has a special section that is dedicated to software development to use. All right. So it's a subsection of a device, or it's a totally independent.
It is a device, and if it drives the surgical decision, the surgical act is a class 3.
All right.
It's extremely tough.
Michele, do you remember?
And I like that you said, I really liked the expression regulatory scientist. It's the first time that I hear that. But it's true, because this is a spectacular expertise to have. And I really agree that it should be called regulatory scientist.
Yeah, that's the way they are called. Yes, that's the way we should be calling also then. All right. All right.
All right. So thank you for this actually great roundtable. I didn't expect it, so thank you. Well, I'm also very glad that we are perfectly on time, so we go for lunch. I thank the speakers again for their time
Where is the lunch? Where is the lunch?
In your canteen, I don't know. Go in a small place here. Yeah.
Unbelievable. Unbelievable.
Yeah, personal lunch, and see you at 2
See
p.m.
you in a bit. See you in a
See you in a
bit.
bit.
Thank you.
Good
Bye-bye.
night.
Bye-bye.
Bye.
All right. Ciao, Martina.
Here we are. Yeah. Wait.
Yeah. You have multiple microphones open in the same room. I was actually going to drop an email because I was waiting to be admitted. I guess the very long and intense discussion over the lunch break are taking us. Can I admit people? I cannot hear you anymore. Is that my problem? No, Martina, you are muted. There are other microphones in the same room that are actually active, and that's the echo, the origin of the echo.
Okay.
Sounds good. Ciao, Paolo.
Ciao Paolo, eccoci di nuovo. How many?
Go ahead then.
We are 60. I would like to wait a few minutes to allow more people to join the session, if you don't mind.
That's perfectly fine with me. I was actually wondering if I was actually connecting to a different room, because in addition to the two major questions that Michele has asked early this morning, so can you hear me and can you see my slides? The additional question is, am I in the right meeting?
Yes.
That has been you know, quite interesting over the last 15 months or so.
I think so yeah, let's wait 3 minutes more.
Sure.
Thanks.
So Paula, this is an hybrid event, right? We have a number of people that are also there in the auditorium, in the room with you, right?
Yeah, it's a lot. The people of the team mostly, and the colleagues from Politecnico, and the colleague from Mario Negri, because it's just here.
Paolo.
Yeah, Paolo. Ah, okay, he's here.
I was actually wondering because I haven't seen him written anywhere on the screen, so I assumed that he was in presence there. And yeah, ciao Paolo.
I'm connecting with my computer, and so I spend a lot of time for the
There you go.
technical reasons. From technology to biology, and to biologist, and not so much technologist. So this is the problem.
The biology is working a little bit better.
Yeah, maybe.
Ciao. Couple of minutes and we start.
And I guess also Filippo Rossi is in the auditorium with you guys, is that right?
Yeah, yes. Come vuoi? Per me va bene, poi mi devi passare la presentazione. Oppure, come preferisci tu? Sì, va bene. Yeah.
Stephane, what is the situation and Michele, what is the current situation in France? Are you allowed to have meetings and events of a smaller size under controlled conditions in your universities? Because France has only recently reopened the borders, right? You have been shut for quite a long time.
I have to say that we are back to almost a normal situation. We are hosting meetings here in physical. Today there is a urology course. There are like 20 surgeons because we do also hands-on training. Now it's going quite well. And since yesterday, they have extended the couvre-feu. They have extended the time you can spend outside up to 11 p.m. And there is a reopening of the bars and restaurants. So good times. Sure.
No, same thing here. I don't know. I think it was until 11 p.m., but now they extended also farther. So I've seen that, for instance, some bars and grocery stores are open until midnight. So I guess, but Italy, it goes by a region. So every region might have different conditions, and I guess it's the same also in France. Well, looking forward to have this meeting, perhaps sometimes next year in presence at this point, across the Alps, or perhaps still in Italy. We'll see what Paolo can do.
Yeah, sure.
Sorry, I was talking with.
No worries. We were talking about you, but only good things. Just loading you with additional work.
Yeah, to stop the pandemic by.
That's all I would say.
No,
Okay.
the beauty to be in a medical environment is also that most of the urologists that are taking the classes and the course right now are probably multiple time vaccinated, right? So that also limits and facilitates the process. All right, Paolo.
Yeah, we can start. We start this afternoon session on nanomedicine with Professor Decuzzi. You can share your screen.
Getting there.
Perché? Perché quello che ho condiviso.
All right. I'm not sure if you're seeing the right format, right? Do you see a full screen presentation or not?
Right now we don't see anything.
I was actually kicked out from the presentation. How about now?
No, yes.
It's coming up. It's coming up.
Yeah.
All right.
Yeah.
So let's see if it moves. Okay, it does move back and forth, right? You see also the other slides. All right. So whenever you want, I'm ready to go, Paolo.
Please go. Thank you.
All right, fantastic. Thank you very much, and welcome to the afternoon session. You will see that there is a little bit of a change in speed and gear here. in the sense that the first part of the morning was mostly focused on, let's say, macroscopic technique. We have seen how beautifully mathematics and computational modeling can be applied to solve practical problems and advanced our surgical techniques. We have seen imaging and advanced, of course, surgery tools. And in the afternoon, we are kind of zooming more down in size, and we're going to into the nano and micro world. I will basically present a couple of different technologies that are more on the nano and micro size. Two examples of what we do in the lab. The word nano, of course, is it has been around for a number of years now. I don't know why, but the screen keep on changing, and I hope that you are basically still visualizing what I do see. I'm visualizing what I do see. And the word nano has been cool for a number of years. And then more recently, the number of nano-skepticists has dramatically increased, I should say. And we start listening to people saying, nanomedicine has promised a number of things, but has delivered nothing. Nanomedicine is not really the future. We should look into other things. And then, of course, 15 months ago, an interesting event has affected our lives in different ways. Some lives more dramatically than others. But I guess looking at 15 months after that, we need to start really kind of making a list of the good things the few good things that are coming out of this pandemic, and how to basically continue and build on top of these very odd events. And for sure, one of the things that I would like to highlight, and perhaps has not been highlighted in a very broad and wide manner through the public, is that the reason why we do have now a couple of vaccines that are really working, and they have the highest level of efficacy in terms of preventing infections and also spreading of the infection from one individual to another, is because over the last 10 and plus years, research in the nano drug delivery has been funded. And the particle that you see right now, this lipid nanoparticle, which is pretty much the same used by Moderna and by BioNTech for delivering the two different mRNAs against SARS-CoV-2, are basically the results of a decade of research. These LNPs have been used and approved in clinical trial for a number of oncological applications, but really, in my opinion, document how you developed a tool for solving cancer, for addressing cardiovascular disease, and then for a reason that is, of course, not predictable, you end up using this in a very different context and very effectively. And now we have most likely hundreds of thousands of people in the world, actually probably millions of people in the world, hundreds of millions of people in the world, that have been injected with nanomedicine. Of course, it carries its own mRNA, but it's basically, let's keep in mind, a nanomedicine. So in my opinion, all the nanoskepticism that we have seen over the last years should really be reconsidered. And we need to use this time as an opportunity to convince even more the public that nano is not only cool, but is actually extremely useful for expanding even more the portfolio of technologies that we can use in healthcare. And not only the public, but also the industry and our friends on the other side, the non-academician, the industrial partners and friends that sometimes have been also increasing that skepticism that I was actually referring to. So I basically just would like to say as a general comment that the next 10 years in nano will be extremely interesting. And I think that we're moving from a sort of more chaotic, fast development phase into a more organized consolidation phase, and we will see more and more and more nano product reaching the clinic and eventually treating diseases. Now, the group that I'm running, and I have the pleasure and the honor to run in Genoa at the Italian Institute of Technology, and I've been doing that for the last 5 plus 6 years almost after I came back from the US, has been working on different type of nanomedicines and nanotechnologies. And as you see a line on this yellow ruler, we basically work on things that are very small, like micelles and lipid nanoparticles, polymeric nanoparticles, you know, pretty much the same thing that you have seen with Moderna and with BioNTech, which fall in the 100 and lower, 100 nanometers and lower characteristic hydrodynamic size, and are comparable with LDL, influenza virus, and so on. But we also work with objects that are still within the nano and micro realm, but larger than the conventional nanomedicines. And these are the discoidal polymeric nanoconstructs, which are about 1 micron in size. We have the microplates, which are 10 microns now, and they are injectable only locally. They cannot be systemically injected. And then the micromesh, which is the newborn from the family, and it's an implant that can be used for delivering a number of different therapeutic agents directly from the site of primary tumor resection. So again, a system that cannot be injected systemically. Whatever is smaller than 1-2 micron can be injected systemically, especially if it is deformable. Whatever is larger cannot. The way we develop these tools is by combining together expertise that belong to the computational science arena. So we do a lot of in silico 3D modeling, different from the one that has been shown earlier this morning, and that probably also the speaker that is following me will be presenting. We're looking at mesoscale, molecular dynamics, and macro scale full finite element methods with a little bit of artificial intelligence at times, but really it's our core solution of equations that are governing the physical and chemical problem. We do a lot of microfluidics, mostly for assessing the quality of the particles. And then, of course, we do also the in vivo, some in vivo. We do collaborate with a number of great scientists all over the world when it comes down to testing our tools in more sophisticated animal models. But we do have a portfolio of different animal models, especially cancer models, in the in IIT in Genoa that we operate with daily, basically. Today, I'll be in the next 20 minutes or so, I'll be focusing mostly on the 2 extremes. So I'll give you an example of a new nanomedicine, a new nanoparticle that we have been developing, which has about 200 nanometer in size. And then I'll be talking about the micromesh, so the other extreme of the if you want, the ruler, with something that can be only implanted locally. So the first particle that I would like to discuss and present to you, and this is unpublished work that has been developed mostly by Roberto Palomba in the lab, and I like to present sometimes also unpublished work, although it's under review. And these are what we call the methyl palmitate nanoparticles, and it's a very simple system, and as you can see, 200 nanometer in size, and is obtained by mixing together methyl palmitate, which is basically a lipid, and albumin, both serum albumin, bovine, human albumin, no difference. By mixing them in proper ratio, you can get basically these nanoparticles, which appear quite spherical in shape with a very good monodispersion and a PDI, which is in the order of 0.1, so very nicely controlled. You can produce them by different methods, either by shaking and baking, a classical homogenization process, mixing, or also through microfluidic system. You need to be very careful in modulating the ratio between the albumin and the methyl palmitate, because otherwise you won't form any particle. You will form aggregates that are not proper in size distribution and in stability. But if you hit the right ratio, then you can get something that is actually quite stable. And we have identified that ratio as being excess of methyl palmitate over serum albumin, as you see basically in the plot at the very bottom right corner of the slide. What do we do with this particle? Is there any therapeutic efficacy of these particles? Well, methyl palmitate has some intrinsic properties, anti-inflammatory properties, antifibrotic properties, and so on. But in the current configuration, we are not really exploiting the intrinsic therapeutic properties of methyl palmitate. But what we discover is that these particles can be used for conditioning macrophages in different organs like the liver, the spleen, mostly, and make sure that the macrophages are basically for a relatively short period of time blocked in their phagocytic properties, so that if we inject a secondary particle, which is actually the therapeutic or the imaging particle, we can basically change the biodistribution of this secondary particle and make sure that it reaches the disease target, so for instance the cancer, more efficiently than if it wasn't if there wasn't any pre-injection or pre-treatment with the methyl palmitate nanoparticle. So it's a particle that we use to put temporarily to sleep the macrophages, especially in the liver, in the spleen. And we demonstrated that in vitro and in vivo using a cohort of different nanoparticles. We have been playing with large particles, we have been playing with small particles, particles that would normally lodge into the liver and in the lungs, like the rigid DPNs, and other particles that would get on the other end to the tumor. And when you do a number of characterizations in vitro and using different techniques, using confocal microscopy on one side, flow cytometry on the other side, and incubating these particles with the cells, and in particular with bone marrow derived monocytes, which are basically very active macrophages, primary macrophages extracted from the femur of a rat, then you see basically that you first expose the cells to the methyl palmitate nanoparticles, and after sometimes a couple of hours, you basically expose the same cells to the nanoparticle of interest. And you see, when you do confocal microscopy, is that before exposure, this was the internalization of the secondary nanoparticle, and as you pre-treat the cells with the methyl palmitate, you basically dramatically reduce the internalization propensity of these cells. This happened for the 750 spherical nanoparticles, for the rigid 1 micron DPNs, for the spherical 200 nanometers particles, for the spherical, what we call SPNs, which are polymer-based nanoparticles, and also for liposomes. This is the case of the confocal microscopy. This is the case of flow cytometry for the different populations and cohort of nanoparticles. You see that the process is concentration dependent, where the concentration is related to the amount of methyl palmitate that you are using. But the most important data, in our opinion, is also the fact that you can recover the internalization propensity of the particle after about 16 hours. So after about 16 hours overnight, let's say, these cells can be normally, start uptaking the nanoparticle of interest. So this dormancy induced into the macrophages is only transient. Of course, it's particle dependent, but it's only transient and can be restored generally overnight. The mechanism is related to an instability of the microtubules. So basically, you can see here also in green, it's much better than the EM, the cryo-EM images. You see that basically there is some background noise. Perhaps if we can mute the microphone, that will be also helpful for me. The control experiment, methyl palmitate exposed to the cells exposed to methyl palmitate, and then after 24 hours, you clearly see that after the treatment, a few hours post-treatment, you see that the microtubule and the cytoskeletal structure of the cells is somehow compromised. So even their ability to deform, invaginate cells and nanoparticles is compromised, but then it's fully recovered after 24 hours. And you can see also from the bar charts that this U-shape distribution of the bar charts is actually indicating that there is a basically full recover or the control conditions after a few hours post-treatment. This is in vitro. What happens in vivo? Well, in vivo, if we take an animal and we inject a ×0 methyl palmitate nanoparticles into this animal, the particles will end up, of course, in the liver, in the spleen, in the major RES organs, and they will start exerting this dormancy, transient dormancy effect. And if you then go back after a few hours and you start injecting rigid micrometer particles that would end up mostly into the liver and a little bit in the lungs, but of course the first organ that it would eat is also the liver, what you see is basically that you can somehow reverse the accumulation of these particles from being mostly into the liver, being also into the lungs. And what is interesting is that after 4 hours, you see the largest ratio between lungs to versus liver. And if you wait for 16 hours, so you inject then the particles, the rigid EPNs, after 16 hours overnight, after 16 hours post the treatment with the methyl palmitate, you basically recover the same control condition with a lungs to liver ratio, which is in the order of 1.5. But otherwise, the effect of the methyl palmitate is very strong within the first 2 hours. Now, this is interesting when it comes to biodistribution. It might be an interesting experiment, but what it comes, really what is important to us is that you can reverse the accumulation of particles and when you have a disease developing into your animal. So here in this case, we have a tumor that is growing in the flank of the animal, and we start treating this tumor using nanoparticles, conventional spherical polymeric nanoparticles that are loaded with docetaxel, 200 nanometers in size. And basically we start injecting these particles after some times, and we follow the size and the growth of the tumor over time using bioluminescence. And what we realize is that if we use the pre-treatment with methyl palmitate, the efficacy of the particles in slowing down the progression of the tumor is dramatically enhanced. If we don't use the methyl palmitate nanoparticles, the nanoparticles can do something, but really there is no almost any difference compared to the untreated. And of course, the methyl palmitate nanoparticles by themselves, they don't do much. If you use PET imaging to quantify the amount of particles that are actually getting into the tumor, the control experiments is the green bar, and the blue bar represents the amount of nanoparticles that have reached the tumor 4 hours post pre-treatment with the methyl palmitate nanoparticles, and you can see there is a dramatic increase in the accumulation into the tumor, which is correlating then with the higher efficacy that we have seen. So this is a system that we are continuing to test using Doxil, using other commercially available nanoparticles, and the idea would be that of doing a sort of pre-treatment on the patient using materials which are basically natural, I mean albumin and methyl palmitate, and preconditioning at this point the macrophages in a very specific and transient manner, so that then when we inject the nanomedicine, we can have a higher accumulation of this nanomedicine exactly within the tumor tissue and within the disease site. This is the first story that I wanted to share with you, and the second story, I think I have a few more minutes, is related to the use of the new device that we call the micromesh for the treatment of brain cancer. Now, this is a system that is macroscopic in size and can only be deployed directly into the tumor cavity, cannot be injected. Glioblastoma, you all know, it's a very it's a very aggressive cancer, the most aggressive brain cancer, a cancer for which we haven't seen any success in treatment over the last 30 years, I should say. There are some new kids on the block in terms of therapy, like Optune from the Israeli Institute of Technology, IFA, that are providing a few months of increase in overall survival. There are immunotherapies that have been basically failing in demonstrating any advantage over the current therapies, which continues to be TMZ, temozolomide, a drug that has been approved in the late 90s with radiotherapy following, of course, major massive tumor removal from the cranium. Now, the system that we're proposing and that we have been recently describing in a paper that came out at the beginning of the spring in April is basically this micromesh. Here in this cartoon is somehow represented as a very well organized grid in green. It's made out of PLGA. These tiny filaments of PLGA are 3 by 5 micron in size. And they are loaded with an hydrophobic drug, which in the current configuration is called diclofenac. So it's an anti-inflammatory, COX-1 inhibitor mostly. This grid is deposited over a microlayer of PVA, which is another biodegradable and actually water-resorbable polymer. The microlayer is a few hundreds of micron thick and is loaded in the current configuration with docetaxel-carrying nanomedicines. So basically, we have two different therapeutic agents. One is a small molecule hydrophobic that is dispersed into this grid, which is actually the mesh, and then we have nanomedicines loaded with a potent chemotherapeutic agent that is basically distributed within the PVA layer. Here you see a cartoon with some dimensions. Here you see a confocal microscopy images showing the PLGA strains in green, the red fluorescent nanoparticles. You can see the punctuated red in the background, and then an EM image that shows basically the border of the mesh and the particles that are coming out from a partially dissolved PVA. The thickness of these strands of PLGA is very small, so this is a very flexible system, and also the size of these openings, square openings of about 20 microns, is comparable with the characteristic size of a couple of tumor cells. And these data are extremely important because you see here an EM image of the micromesh that is being fully released from the PVA layer. This is a fluorescent image of the same micromesh. It really looked like a mesh of a net of a fisherman. So this was actually also called micromesh. We are in Genova, very close to the sea, so it really represents to us, a fisherman net. Fabrication process is relatively straightforward, no time to discuss that. The way it works and the way we have been using it is by positioning it within the resected cavity. And what happens is the micromesh basically interacts with the borders of the tumor, with the margins of the tumor. It basically starts dissolving. The PVA start to dissolve. It dissolves in water. It releases the nanomedicines that are basically distributed within the PVA, and as it dissolves, it frees the grid, the mesh that basically conforms to the surface of the resected cavity, and it starts then releasing the smaller green dots, which is basically the diclofenac. So you have a sustained release of nanomedicine loaded with a very potent chemotherapeutic drug that will not be able to cross the blood-brain barrier, but in this case we are bypassing, of course, the blood-brain barrier, and then we have the diclofenac, the anti-inflammatory drug that is continuously released. The beauty of the system, beside the fact that being two compartments, and so being able to handle two different drugs with two different, completely different profiles, is also the ability of these very tiny strands to somehow conform to the resected cavity as a fisherman net, capturing in these images here a tumor spheroid of a few hundreds of microns and wrap around it as a glove and deliver immediately over it the therapeutic agent. So this is extremely important because this ability to conform to the disease surface increases the potential of the therapeutic agents. I don't have the time to go through the release profiles, but we can actually modulate the release profiles at will. The interactions of the micromesh with the tumor tissue allows us to release for several days continuously both the small molecules as well as the nanomedicines. And when it comes down to the in vivo experiments, we basically were able to demonstrate and show that in a tumor model where the tumor is not being removed, the micromesh outperforms all the different control groups very nicely and efficiently. These are 9 different control groups, 10 animals per control groups, 270 days of observation. We had to basically sacrifice the animals surviving here because of the length of the experiment. 270 days is 9 months, which in the life of a little mouse is a lot of decades, basically. If we remove the tumor mass, which is what a neurosurgeon would do quite immediately, you see that basically there is an increase, a dramatic increase in survival as compared to standard TMZ, as compared to LSPNs. What is LSPNs? This is very important. LSPNs are the same nanoparticles, the same docetaxel nanomedicine that are loaded in the micromesh, which are basically instilled in the cavity of the tumor one time. And even if it is the same nanoparticles, they are not replicating the results of the micromesh because the micromesh is able to conform to the surface and contain locally the therapeutic agents, rather than having a therapeutic agent that is injected in the cavity and goes pretty much everywhere in addition to the tumor margins. And this is a problem that, you know, affects also the injectable gels. The fact that, you know, the drug that you put into the gel, you never know if it is going to the tumor or if it is going also other places. And then if you use primary cells coming from our friends at the San Raffaele Institute that have been extracted and characterized from patients carrying glioblastoma, then you basically see again that the micromesh outperforms the controls. These are the stories that I wanted to share with you. We are continuing developing these stories. For instance, this is a case of a lipid nanoparticle. So the famous docetaxel nanoparticles can be changed with any type of nanoparticle. This is a lipid nanoparticles, pretty much similar to the BioNTech Moderna nanoparticles that is loaded with 11 different miRNAs that had a potential to slow down the progression of the disease. And we actually showed just with the miRNAs that there was an increase in survival. We are not adding any chemotherapeutic agent here. It's just a combination of the 11 miRNAs. This is basically the story that I wanted to share with you. I hope I didn't go too much of time. And of course, I've been presenting this story, but this story is the result of a number of years about 5 years that we have been spending in Genova with PhD students, postdoc, and senior scientists that have been collaborating at each single piece of the story that I just presented to you, and other stories that I didn't have the time to share with you, but hopefully in the near future I will. Thank you very much. Ready for questions or for listening to other talks.
Thank you, Paolo. It was really inspiring. I see a question, yeah, from Somaye, and then we have a question from Eric from the Prof. So Somaye, go ahead.
Well, thank you very much. Thank you for your presentation. I have just one question. I'm sorry, maybe you explained about that, but I didn't understand about the platform made of PLGA, the green color. You referred to the BB, and I didn't understand very well how these nanoparticles or this platform can overcome the challenge of this BB. If you could explain a little bit more, I would be thankful.
Well, the nanoparticles are released directly within the tumor cavity on the margins of the tumor. They're not systemically injected. And so the blood-brain barrier, not coming the nanoparticles from the systemic injection, is somehow bypassed.
So what is your suggestion about the different tumors with more strong BB? I mean, for example, the brain tumor that has a very strong BB. Is it possible to deliver these nanoparticles to this kind of tumor? Because I know that this barrier is very strong and it's very difficult to deliver the nanoparticles or drug.
Yeah, that's exactly the case, Somaye. The major application is for brain tumors, for glioblastoma. But as you might have alluded to, we're also working on different tumors. But yes, definitely, the main application is with brain tumor. And as I showed in some of the slides, we have different brain tumor models where you can actually remove the primary mass, or you can actually leave the primary mass there. But the platform, and I thank you for this question because it allows me to expand a little bit more and provide additional details. The platforms can be used also in other contexts. We can use it on, and we're looking to use it on breast cancer, where there is also a significant resection of the tumor, but limited amount of mass that you can resect, and then on colorectal cancer. But potentially any other tumor where there is a resection of the primary mass that is needed and required, and where there is a limitation on the amount of mass that you can remove because perhaps you are very close to an artery, right? So you are very close to a main blood vessel, or you are very close to a nerve, and you don't want to risk to interrupt the blood flow, you don't want to risk to interrupt the nervous connection, then one way to use the system could be to remove as much as possible, and then on the margins directly apply this system, which is very much conformable and has a number of advantages over tumor cavity injected gels.
Okay. And have you tried about the different kind of the tumor? I mean, that's the tumor with the high grade and the bigger size regarding the high concentration at the deep part of the or internal part of them. Maybe it is difficult to, the biggest challenge is delivering the nanoparticles into the deep and internal parts of the very high grade or big tumor. And can we try, I mean, that these nanoparticles can easily go and penetrate into the deep part of the tumor and overcome the high pressure in the internal part of the tumor?
Yeah, so we started looking into this, and I just ran through the slide, but basically we show that the same nanoparticles, and by the way, the nanoparticles that we are using are very simple. And I mean, we can use any type of nanoparticles. It could be liposomes, it could be a lipid nanoparticle, and so on. What we showed in vitro using tumor spheroids, which are very compact, they're actually more compact than the actual tissue in the tumor. We showed that using the particles released from the micromesh, this can travel deeper into the spheroid as compared to the same nanoparticles that are not being released from the micromesh. So there is an advantage, and I can tell you that this advantage might be related to the alteration of the surface properties of the nanoparticles which are released from the micromesh. We are continuing doing experiments on these. We have done also some intravital microscopy, preliminary intravital microscopy data, and I apologize, the slide was there, but I had to run over it. But I will be happy to discuss this further also offline, Somaye, or Somaye, I'm sorry if I'm mispronouncing your name. But yeah, we are continuously investigating that. But shortly, I can say that there is an advantage in using the micromesh for improving the penetration depth of nanoparticles as compared to the case with no micromesh.
Yeah, but yes, you're right. Thank you very much for your explanation. Maybe it would be very valuable to test it in the solid tumor, the big tumor, to check the penetration in the deep, because it is a very big challenge. Thank you very much for your explanation. It was great. Thank you. Thank you.
Thank you. Thank you for your question.
Thank you, Somaye. Thank you, Paolo. Paolo, the next question I've asked the...
we can take it later.
Yeah, later,
No,
exactly.
Definitely. All right. Thank you.
Very much. Thank you. And now it's my pleasure to leave the floor to Professor Rossi. Filippo?
Yeah, we are in the same room, but quite distant. I share the screen. I hope that you are saying and not listening to me, otherwise, please tell me. Okay, good afternoon, and thank you, Paola and Martina, for this wonderful organization and this wonderful opportunity. Following the talk of Professor Decuzzi, I share with you the sum of the experiment we did and we are now doing in Milan on personalized targeting and drug delivery. If we consider drug delivery and the targeting is only a specific part, we have different kind of opportunity. We can be interested in enhance the penetration, in controllable release, so for on long kinetics for long time, to extend the circulation, etc. Today, we will focus, we are doing many of these different works in my group, but today we will focus our attention in the increase of cell uptake in order to improve the selectivity of nanomaterials. Basically, we can imagine that they can work like Trojan horses so they can carry and enter selectively within specific cells, carrying and releasing their content only in that specific cells. But start with the medical problem. The main application we are working with, due to a strong collaboration with Mario Negri Institute for Pharmacological Research here in Milan and the group of Dr. Veglianese, is on spinal cord injury. Spinal cord injury is the result of a primary event, that is the contusive stretch injury, like car accident, motorbike accident, for example, and so on. That is then followed by a secondary one, that is commonly known like the main cause of the post-traumatic neurodegeneration. In the European Union, every year, more than 10,000 people suffer from spinal cord injury, leaving permanent neurological diseases in almost all 100% of the cases. We have also to consider that the average age of the patient is generally quite low than 30 years old, so there's a lot of cost in terms of society, in terms of economic, and in the life in the everyday life of this patient. Nowadays, there are no solving therapies available, but the research community is working, in order to provide drug or cell delivery, trying to improve the medical chances. What does it mean? Just a brief about spinal cord injury. It's true that I'm a chemical engineer working on chemists, so I'm not mostly skilled about that, but it's just an introduction before we can then discuss about the material part. We start with a primary injury. So we have synthesis of nitric oxide, oxidative stress, microglia activation, local inflammation. All these phenomena then cause many other phenomena. So it's a cascade of phenomena that happen at different steps also in terms of kinetics, not at the same time. But you can see that we have neural necrosis, neural apoptosis, scar and cyst formation, demyelination and disruption of morphofunctional nerve pathways. So the main aim on one side is to counteract the mechanism of secondary injury and minimize the pathological consequences, while neuroregeneration will try to rebuild the damaged tissue. Starting from this is a picture of what happened after the mechanical damage in you can see in picture in one, the mechanical damage. We have the resting microglia that become activated. There's also the infiltration of macrophage that enter within the central nervous system, and they cannot be easily distinguished with the activated microglia. And they have two phenotypes. A good phenotype that release anti-inflammatory agent is named M2 phenotype that happens at the beginning. But after a certain time, when the system look that the damage is too huge, is not able to be repaired, work in the opposite way in M1 phenotype. M1 phenotype release pro-inflammatory agent and exacerbate the damage. In cascade, also astrocytes are activated, they create a glial scar, and we have also the loss of neural apoptosis and necrosis due to the release of pro-inflammatory drugs, both from microglia and activated astrocytes. So our system, if we consider from a biological perspective, the actor of this pathology are most the one we listed. We have a microglia, astrocytes, and neuron. We started almost 10 years ago trying to say if it would be possible to release selectively a drug within a specific cells. And first of all, we consider microglia. Microglia is macrophagic cells, and we started to use nanomedicine, so polymeric nanoparticle, to selectively release drug within microglia. We considered different kind of nanoparticle. The most promising one was the one where the synthesis is presented here. It is an hydroxyethyl methacrylate. It is a nanoparticle synthesized by emulsion polymerization. We start with the formation of a macromonomer with the addiction between hydroxyethyl methacrylate and caprolactone. That is a cyclic ester commonly used also in medical devices. We have a ring opening polymerization that takes place in bulk, and we can form the macromonomer. We then added an hydroxyethyl methacrylate with PEG. The two vinyl group present at the end of the macromonomer and the two monomer basically in emulsion take place, and we were able to obtain nanoparticle already in colloid state, so in liquid-like continuous phase. We have different possibility. The advantage of this system, but we can have quite high tunability, because changing the number of caprolactone, we can increase or decrease the dimension of the particle. Basically, if we increase the n value, we decrease the dimension of the nanoparticle because we increase the hydrophobicity, and vice versa on the opposite side. We can see from TM analysis and the DLS the dimension around 100 nanometer. Then we started to study the selectivity of this system. We use primary co-culture of microglia, astrocyte, and neuron. We introduce an inflammatory stimulus, lipopolysaccharide, and so we can divide LPS cells, so cells that are activated, and control that are resting microglia. We added our nanoparticle, and what we can see from the confocal microscopy, that this kind of nanoparticles are uptaken only by microglia, especially by the activated one, that is of course more responsive to this kind of external stimuli. From the both, I don't have the pointer here, but from the picture on the right side below, you can see in the red the polymeric nanoparticle, while in green you can see the membrane, in blue the nucleus, and in red all these kind of nanoparticles that are uptaken. Looking to the other cell lines, we were not be able to see any sort of uptake, underlying that this kind of nanoparticles are selectively uptaken by microglia. We then continue the characterization of this system, looking about the time needed for cell uptake, and we see that the uptake take very quickly in just in the first hour, and then with a plateau after 2 days. The higher selectivity on the right upright graph, it is visible. So these kind of nanoparticles are uptaken mainly by activated microglia. And then from classic in vitro analysis, we also demonstrated that this nano object did not affect the viability and cytokine release with MTT and ELI assays up to 6 days after the internalization. We also decided to study a little bit, not too in details because it is quite far from the final application, because we know that there's a huge field of research that work on the mechanism of cell uptake. We know that some kind of materials, some kind of objects, some kind of shapes, because also the shape is very important, are uptaken by some cells or not. The mechanism behind are many and many complicated to understand. Also, especially from our side, that we're more interested in the final application. But basically, we can see that the main reason of this cell uptake is due to endocytosis. This is why we performed the same experiment we did before, introducing chloropromazine. Chloropromazine is an inhibitor of endocytosis, and we can see from the graph that increasing the concentration of chloropromazine will decrease the cell uptake. Underline, but not until 0. Some of them are already uptaken. So underline that endocytosis is probably the main mechanism of cell uptake, but also many other are interested. This is some we told many times is needed more details, but we hope we can find a good collaboration in the future with groups that work in this way. From TM analysis on the left, we can see that this kind of nanoparticles are then enter within lysosomes, and they are then degraded after 6 days. We then loaded a drug within this kind of nanoparticle. We use minocycline. Minocycline is a well-known anti-inflammatory drug, and here are presented the results of the treatment of control, meaning resting microglia. LPS is activated microglia, so in a round shape in a phagocytic condition. And we can see that introducing minocycline without nanoparticle, we can see only a partial deactivation, while if we introduce minocycline within nanoparticle at the same concentration, we can see a complete, a higher degree of deactivation of microglia, visible also from the time-lapse analysis in the picture, when the same cells start with round shape at 0 hour and then become elongated after a few days. And so we can see that the selective treatment can reduce the inflammation of microglial cells. It doesn't mean that we can shift from M1 to M2, because minocycline is not able to do that, but we shift from M1 condition to a neutral condition. We also perform the same analysis in vivo, and so we inject within the parenchyma nanoparticle loaded within minocycline, and we can see that only in the case of minocycline loaded within nanoparticle, we can see a good the activation, underlining that this kind of selectivity is possible not only in vitro, but also in vivo. And after that, we perform some behavioral studies in order to see the amelioration in terms of behavioral outcome that can take place in spinal cord injury mouse model. We injected this nanoparticle suspension in liquid solution with precision with microsurgery, and we can see from Basso mouse score value, that is a value that represents the quality of the walking ability, so the ability to move to mice, the amelioration that is possible with this kind of release. We also studied, so on the left side, we can see that one of the other thing that we can see is that this kind of amelioration is possible only if we deliver, if we make this injection next to the accident, in this case to the injury of the spinal cord, while if we inject after 1 day or after 7 days, named T1 and T7, this kind of amelioration decreases due to the fact that minocycline this is a problem, if we can see, of the drug, that it is effective only in the early stage. After that, after some times, we decided to combine, because we have a sort of amelioration. I forgot to say that this mouse score in mice is between 0 and 9. 9 is an healthy animal that moves. We can reach a plateau value after 28 days and after more than 1 month of 4. So we have an amelioration, but it is not enough. It's not enough because we said that this kind of injury is a multi-injury. So we work against only specific cells. So this is why we decided to work and to fight, this is kind of picture, against astrocytes, another category of cells, extremely more difficult to, to treat due to the fact that microglia is phagocytic, so more responsive to external stimuli, respect to astrocytes. In this case, we developed another category of nanoparticle based on PEG-PEI. So we modify the PEG with carbonidimidazole, forming the PEG-CDI. We then combine PEG-CDI with polyethylene imine, present in the second reaction. This polyethylene imine can be functionalized with a click reaction with chromophore, in this case rhodamine, but also with CY5 for in vivo analysis. So we basically functionalize the two polymers. So the first one with CDI, the second one with the chromophore, and then we combine with an emulsification evaporation procedure, and we can obtain the nano object. this nano object is bring together by carbon-mate bond that are visible around here, but we have also some region of potential polymer functionalization. In order to develop this kind of nanoparticle, we decided to change dramatically respect to polycaprolactone and this kind of polyester, because it's commonly known in literature that they are extremely responsive only to microglia and macrophage. And in the development of this new family of nano objects, we also look about the versatility that we can have. So we can see that there are some dots, some point of functionalization that can be then functionalize and change in order to address different kind of needs. The different kind of needs, we can say, can be a little bit listed and presented like in this graph. So we started with PEG-PEI nanogel. We introduced some post-polymer functionalization strategy in order to have a versatile family that can work in controlled drug release possibility, and in this way we studied the possibility to deliver a drug carried by electrostatic interaction between the polymer chain and the drug, or the sulfide bridge. But today we said that we will focus our attention on cell interaction, so we introduce some other coatings. So we maintain the main structure of the system. We introduce some small molecule on the surface with the final aim to direct and to target their delivery. Then if we have a couple of minutes left, we can also say something about one of the two other possibilities. In this direction, we can back again to the same extremely simple reaction. Even if we are fascinated by chemistry, we are fascinated by polymerization, we decided to make them simple as possible, for reasons that in this audience are very easy to understand. So we start with PEG-activated imidazole, PEG-conjugated CY5, in this case for in vivo analysis, and we obtain a nano object. This nano object can be then decorated with specific moieties that can direct their uptake and improve the selectivity. We tried many of them. As in every oral talk, I'm saying these things mostly for the students that are listening to this piece of work. We are, it is valid mostly from my side, I'm talking about a linear, it seems to be a linear story. So we did this and we obtained that. We did this. This is not so linear. So we tried many, not random, not more in a more engineering way as possible, but after some trial, looking about what literature said, because again, remember that literature is really important, so knowing what the other are doing, it is fundamental for doing a good research. We introduce, so in this kind of nano object is really small, so it cannot be exactly named like a coating because it is only a small molecule, it's bromopapagilamine, so it is only an H2, CH3, CH3, so C3, H9 bromide, that then the bromide is then removed in the reaction. And we can obtain this moiety named visible in red. You can see the round shape of the particle for AFM analysis. The diameter is around 100 nanometer that are stable also in physiological condition for more than 5 days, then they start to degrade. From FDR analysis, only a characterization to understand to prove that the carbon-mate bond present already in the last nanogel and the presence of this kind of decoration that with a good stability. We then treat against co-culture, because of course, this is another important point. To prove the selectivity, we have to work in co-culture in vitro in a more similar condition, more similar as possible, that we can obtain in vivo. We treated with LPS, so we can induce a pro-inflammatory phenotype, both in astrocytes and microglia. After 24-hour exposure of cells of this kind of nano-object, we can see a large, from red dots in the up left figure, so a large amount of nanogel in astrocyte, a few internalization in microglia, and a non-internalization in neuron. This is very, very important. It is quite commonly known that neuron is a type of cells that do not easily have uptake mechanism with nano-object, but the main aim is that we can can insert mostly in astrocyte respect to microglia, and this is very important. The decreasing then after 5 days demonstrated the degradation that we also seen in the previous slide, and comparing with the polycaprolactone nanoparticle that are the nanoparticle we consider in the first part of this talk, that we said present specific and high selectivity with microglia. We can see that with kind of nanogel named NG here, we can drastically reduce this uptake, and so some of them can enter within astrocyte, while polycaprolactone-based nanoparticles are not able to enter within astrocyte, but enter only within microglia. Again, very quickly, we tried here to understand the mechanism of in a very simple condition with chloropromazine, that is a clathrin-mediated endocytosis inhibitor, that confirmed that here almost we reach 0 value. So this basic endocytosis seems to be the mechanism behind this uptake. And also the indicator in the lower image can confirm the degradation, also in this case by lysosomes of this nano-object, and that only a small number of them enter within microglia, and most of them enter within within astrocytes. And also by the no localization, another information we can obtain by the no localization detected, that only few of them are uptaken and no lysosomal activity is visible in microglia cells. After that, we also tried, because we learned by our experience that it's better to work step by step. We tried at the beginning, and this is valid every time, we tried not with a real drug, but we tried with a drug mimetic in order to be sure that what we are doing and to understand why the results is good or not. And we tried with TOPO3. TOPO3 is a good drug mimetic for small steric hindrance molecule and present a good peculiarity that it cannot enter within cells if it is not carried. So if you see the signal, and also because the fluorescent signal is far from the signal of this kind of nano-object. So it means that if you can see its signal within cells, it means that something like Trojan horses we said at the beginning enter and release within cytosol. In this case, we can see the presence of TOPO3 underline the presence within cells, so nanogel can enter and TOPO3, and the absence of colocalization between the two signals say that this drug can enter using nanogel that can then release their content. We inject in the parenchyma adding rolipram like a drug, and we can see, or we proved also in vivo After 2 days, the internalization done by astrocytes, few by microglia in the lower part of this slide, and known by neurons. From microglia, that it is the H value in the lower part of this picture, you can see that only the few signals underline that in vivo even fewer cells are able to uptake and to internalize this kind of nanoparticles. While the signal in GFP and GFP of astrocytes underline the selectivity with the system. Briefly, because probably we are late, we have also other possibility to decorate with this kind of nanoparticle. One of them that can be useful is to use a disulfide bridge. The use of disulfide bridge is important because this kind of disulfide bond can be cleaved by cells. So it means that if we link our drug with nanoparticle using this kind of chemical bond within and can enter within the cells, we can be sure that this drug is not released before and can be released within the system by the cysteine. The cysteine and the glutathione present within cysteine can break this double bond, so we can have nanogen on one side and the drug released within the system. These are preliminary drugs we did with some of fascinated chemistry that make it very, very complicated. I have to be honest with you, they make it extremely complicated to go further. But basically, we are trying also to reduce and to simplify the sort of chemistry. What it is can be interesting is that we can be sure that this drug is not released before and can release only within the cells. These are the plot on the left side in vitro with the glutathione or without, and in the presence of glutathione, we can see the higher release. And from the right part within microglial cells, in this case, because these nanogen are not decorated with the moiety, so we can high microglia cell uptake, we can see that can enter with this drug mimetic and can release. But I hope we have time in the future to, we are quite late to investigate in detail this other kind of possibility we have. But in general, we can see that we talk about the design of nanosystems that can selectively have a cellular interaction using a coating layer. We can also use selective antibodies and make it complicated. This is why we decided we try to work on coating layer, moiety is added in order to have a selective drug release. The final aim is to build a composite system. So we decided to work separately, and then the combination will be also complicated. It's not easy as it seems to be in order to work on neuroprotective way in one side, but also neuroregenerative. This is the other work we are doing with hydrogel, with stem cells. This is but it is another story, in order to have a complete in vivo outcome of different kind of, things that can, of guns that we can have, can fight against different kind of phenomena. My presentation is finished, but I'd like to acknowledge all the people involved in this work, mostly Mario Negri Institute of Pharmacological Research, where I spend sometimes during in Dr. Vellanese's lab doing in vitro and in vivo studies. Some of the in vitro studies that were done in Cambridge University, and some material characterization and optimization in Uppsala University, ETH, and Keio University. And I thank you all for the patience and for being here. And here are my contact, and I welcome also your question here or in the last session. Thank you.
Thank you, Filippo. Thank you very much. Very, very interesting research. Is there any question from the floor for Professor Rossi? It's better maybe to ask now because you cannot be with us longer afterwards, right?
That is a bit. I can stay. I can stay until the end of the work. I can stay Yeah.
Thank you. Is there any question?
No, so we can do it directly at the end. It's good. It's good for you.
Yeah, fine.
Okay, thank you. Thank you. And now let me introduce.
I'm sorry.
Sorry, let me introduce the last speaker, Dr. Paolo Bigini. Thank you, Paolo, for being here with us today, for closing this workshop. Yeah, I leave the floor to you.
Okay. First of all, can you hear me? Can you hear me? Okay. So maybe you have to switch off. Okay. Thanks. First of all, thanks a lot, Martina and sorry, Paola, for the invitation. And let me say that I very excited to be here to present my results sitting in a real room and with a physical audience, because it's the first time. So just a moment, I have to, yeah, maybe it can work. So just to introduce my last talk, I think it's important to say that in the last couple of decades, a great effort was carried out by different parts, so the physician, engineering, and biologists to try to transfer chemical and physical studies for biochemical sciences. And the results, in particular in preclinical studies and in the understanding of the behavior and interaction between these new devices and the biological matrices, are very interesting. Talking about the passage from nanomed to nanotechnology to nanomedicine, I think that the previous speaker have already described very well in a perfect way which are the main steps to perform this kind of presentation. So I decided to move to this talk, to the last talk, both showing some results and some philosophy of our work in the second part. But at the beginning, I'd like also to, since I work in an Institute of Pharmacology, I also to stress some parts related to the passage from nanomed, so the application of nanotechnology at biological level, to the nanodrugs, or that the production of biological and pharmacological products to be used, like happens in for vaccine, as Paolo Decuzzi showed us before. So first of all, this is a very famous figure, when but it's unfortunately it's still ongoing, because there are a great loss of results and the products that fall in the so-called valley of death. And this is not exclusively to problem related to the technology or biomedical studies, but there are many other parameters that can be accounted for this dichotomy between the production of new devices and the real application to the clinical practice, such as the scalability of products, the costs, the regulatory problems, and the ethical problems, and the safety of the product. In the seventh framework program, European Community Union invested a lot of money with a very large number of projects related to nanotechnology and the application of nanotechnology for biomedical biomedical aims. But unfortunately, all these, almost all, not all, almost all results did not produce a real impact in terms of drug production. And the problems are difficult to solve in few times, but the decision makers decided to change a little bit the philosophy of the work. And so, which is the real and the most ergonomic way to move from nanotech to bed of patients to real application of nanomedicine. There are a lot of possibility, and it's not so trivial. But anyway, one of the most important points is the awareness that something should be done. And for example, there are these two documents, two white paper, published by a European Platform of Technology on the left and European Commission on the right. They were published more or less 5-6 years ago. Then are quite interesting. I suggest everybody, if there is a possibility to check these documents, because are very, very interesting, and they are related to the contribution of nanomedicine to Horizon 2020 and also to Horizon Europe, and an overall strategic vision of research and impact on future nanomedical for future nanomedical products. So I try to pick up some phrases that I think are very interesting and maybe could be also a source for the scope and discussion afterwards. The first, from the first document, clinical translation needs innovation. And the second, innovation needs technology, sorry. So if you merge the two sentences, you can say that clinical translation needs technology. It is a very, very important point. For the reason, starting from Horizon 2020, but also in this new Horizon Europe, there was focusing many projects, and the evaluation of projects were focused on the evaluation of the impact of this research in nanomedicine, but not only in nanomedicine. And I select three. There are many points, but three main points I'd like to share with you some my personal opinion and the pro and the cons about them. The first, to emphasize the technological readiness level. The second, to stress the value of innovation. And the third, to try to accelerate the processes of collaboration between academia and big pharma. In a theoretical point of view, they are perfect, because everybody, I think, wants that the 3 points can be accomplished and achieved, but it's not so easy, in my opinion. So we start with a technological readiness level. Now, everybody, when we start to read the call or the project and so on, we bumped into these familiar words, TRL. So, which is the TRL? First of all, we have to define which is the TRL. The technological readiness level is a measure of risk management, and was first adopted by NASA in 70s, but was adopted to define the advancement of the project over the time. So, taking for a long period of time, they want to measure which was the real advancement on the research. Afterward, this parameter was taken from different other industrial, was used for different industrial project, and was adopted by industries and companies. This is what European Union wrote about TRL. It starts from an idea, or original idea, very often, and moving from, I don't know, the proof of concept, the testing on the right environment, and so on, should arrive at a full commercial application of the product. In my opinion, this parameter, in particular for project of nanotechnology or technology, can be used and adopted in a project where maybe there is a prototype, and then we can check and perform following a step-by-step analysis, the development, if this project works, the scalability of the project, if it can be repeated, and obviously the economic burden and the advantages. Just to, I don't know, in this example, just to move from a new monitor, new screen for watching TV. But everybody knows that the third law, exploiting the third law of thermodynamics, everybody knows that when you increase the complexity of system, in this case moving from fine regulation basic science on fine regulation of parameter and shifting from biology and even better in the clinical practice, the entropy increases. So very often it's difficult to guess, to decide before which will be the increase of complexity of the systems. And I think this is the easier example to show why very often in biomedical, nanomedical, but biomedical in general project, the TRL is a little bit weak. If you consider the total number of drugs that start in the study in pharmacology, more or less, more than 95% of drugs fall in the so-called in vitro phase. For the remaining compounds, even in this case, in general, another 95% could not pass the preclinical phase. So starting from, in this case, 10,000 compounds, we can arrive at level of 6, 7. So about what in many projects is required. A very low percentage of compounds. So it's very difficult to say and to understand that before starting. We can know which is the starting point, but it's very difficult, and very often this is a great limitation in terms of project. Another important point is innovation and validation. Everybody knows that this is an important rule to move from the possible development of drugs and other biomedical devices. And this is another sentence taken from these documents. Competitive stem from technological innovation. Biomedical projects, in particular in nanomedicine, have been tightly connected to the generation of innovative products, right? And higher innovation should lead a greater impact. Everything is true, but unfortunately, for an unavoidable reason, technological innovation, such as the production of devices, particles, and so on, run manifold faster than biomedical translation. To evaluate in a serious way the impact, as Filippo and Paolo before showed in biological matters, require a lot of time. And so very often there is a poor characterization during the project of this bio-nano interaction in terms of stability of compounds, kinetics, and safety, just before looking at the efficacy of the compound. And in terms of potential impact on potential development to the for practical use in patients, this is actually generate a lack of robustness of data and the waste of time, money, and very often material. So a possible new way to combine innovation and validation could be exploit a first step in which a basic project can be performed to select the best, in this case, engineered nanomaterial, but other type of devices by exploiting by proof of concept. And in a second time, for example, in the same framework project or in the other project, it will be possible to more carefully study the selected project for other granted project, and in order to have a fine characterization of the interaction, increase the robustness and reliability of of results in order to improve also the trust in general, the trust in nanomedicine, and in general in innovative medicine. The third point is the interaction between the academia and the big pharma and the industry. This is a very important point because otherwise this is one of the main reasons because many compounds fall in the valley of death. And also in this case, there was an attempt to evaluate which were the point of weakness. In this phrase, they wrote that one of the main hurdles for the translation of nanomed products is that in many cases there is not the possibility to generate a contract manufacturing organization that can be started to proceed for the valorization and exploitation, technological exploitation, and also the condition to make them scalable for the production, for the large scale production. This is true, but also in this case there is a formal problem because very often the research project granted for many associations and agencies require dissemination, and dissemination is a most important point also for the career, academic career. So it means publication, the spread and the sharing of know-how, and also an increase of social and cultural level. And this is good actually. But unfortunately, very often the impact it's a little bit different. They want to decrease the mortality and morbidity in terms of obviously biomedical products, and industrial and economical development, and decrease in social and economic burden to the patients and so on. But very often they require the results should be secreted, and there are agreement for no disclosure to not share, to have the possibility to maintain the confidentiality of the product. So I think it would be very important to evaluate which, if you want to move, is not exclusively, this connection is not exclusively related to the production of a contract. I think we should a little bit to revise the type of collaboration. And this is what can be done at the end of a single first part of the project in which communication, paper, networking is granted. It's important to try to find some rules, common rules by starting from officer, regulators, industry, and so on, to enable the exploitation and transferability of this data to the philosophy of the industry, so data protection and confidentiality. So in general, I want to just to finish this first part. The application of TRL is not so bad, but maybe should be reconsidered thinking about biological variability. It's not possible to apply the TRL used for the production of other type of products. The innovation is important, but should be combined to the biomedical impact, and very often the 3-4 years long-lasting project. The time is too short to guarantee from innovation, a total innovation of a product, and to arrive to the patient bed. And finally, obviously, the effort taken from decision maker by Horizon are fine in general, but anyway, in this case, as I told you before, it's difficult in using this format to combine the academia needs with the industry interest. And in general, I can say that in my opinion is that this kind of project generate a great value in terms of dissemination, but impact remain a little bit lower. The second part, how many time? 10 minutes. 4 minutes. It's okay, very short. Yeah. Okay. Just to share with you our philosophy of work. I work very close to the here in Politecnico. This is our institute, and in our institute is a pharmacological institute based on pharmacology and global and public health, and there are 4 different departments that are focused on different pathologies, so cardiology, neurology, as Filippo showed before, the collaboration with a group in neuroscience, oncology, and nephrology. These groups are focused on single or a group of pathologies associated with these organs, and we have people involved in preclinical studies with animals and cells, but also people who work in, we haven't patients, but we work in follow-up or post-clinical study, epidemiology, and so on. So there is a great connection between the health, the pharmacological production, and the basic science. I work in a department of molecular biochemistry, and our role is to try to be the link between the different departments and the biological, the basic science that I told you before. And in particular, our activities focus on imaging, nanoimaging, and pharmacokinetics. Just to start to move our research in a similar way than patients, we have set in our building a mouse clinic when it is possible to follow the pathology of in murine model, in rodent model of different pathology at different time points, and also the pharmacological effect of drugs or nanodrugs. In particular, we have the possibility to perform monitoring in living animals, treating them like, or sometimes better, like patients, and following both the pharmacological studies or imaging, in vivo imaging study, and also looking at what was the clinical progression during the treatment. Moreover, since we have the possibility in our small patient, we can also perform experiment with a higher level of resolution, moving from the whole body, passing through organs, cells, and organelles, and it's possible to exploit this part by microscopy, conventional microscopy or confocal microscopy, and nanoscopy and microscopy, so moving to the super resolution imaging and transmission electron microscopy. So there is the possibility to have a very broad range of analysis, to make a very broad range of analysis, and to perform a lot of experiments. So they are the 3 main points. So the drug delivery, stem cell therapy, and diagnosis. We work in this space, more or less with the same philosophy. I show you very briefly in 5 minutes, more or less, 3 different studies we did. First, we performed a study with crystal nanocellulose nanoparticles, fiber, were linked and drug alendronate that can interact with bone. It's used for osteoporosis. And we treated a systematically animal, and we found by in vivo imaging that a signal, in particular in the first day after systemic administration, was in correspondence with bones. Then we treated in purple. This is the purple. It was injected in the muscle, and we found very interesting, using the X-ray in this case, that the nanoparticle, the purple nanoparticle, so the crystal nanocellulose, crystal nanocellulose, can be released and have a great tropism and great interaction with the bones. In this case, at a higher magnification, very close to the vertebral column. And finally, there is the possibility also to perform experiment with micro CT. And in this case, it's possible to follow if this type of nanoparticle can somehow, the chronic treatment can somehow reduce the alteration in the, for example, here in the mouse bones after non-union fracture. So there is the possibility to move from the imaging to the clinical pharmacological application. This is another project. In this project, we perform an experiment using nanoparticle linked with dexamethasone and with a fluorescent dye, so there is theranostics. And we decided to include dexamethasone in nanoparticle before we have the possibility to study the effect of dexamethasone in a model of autoimmune hepatitis. Both in patients and in mice, dexamethasone works and generates an anti-inflammatory effect. But unfortunately, in patients, very often the free formulation, the treatment with the free formulation is interrupted because there are a lot of side effects. In this case, we already knew that nanoparticle arrive most selectively in the liver. Before, Paolo Decuzzi said that there is the possibility to try to pass over the liver. In this case, we wanted that they arrive in the liver. And indeed, we found very, very briefly that the level of drugs with the nanoparticle in this animal released was exclusively in the liver, not in plasma and in brain, and it remained for a longer time. So it was very interesting. But I think most interesting was the results from histopathology of these mice. We found that this is an healthy animals. In brown, in this arrow, there is the feature of inflammation, in particular in this case fibrosis, and the treatment completely revert the fibrotic effect. And here, using a marker currently exploited for human to test human liver health or disease, we found that the transaminase level, this is the control, when we treat the animal with ananas, this nanoparticle, and dexamethasone, decrease, and so there was a clinical effect. And just to finish, we can also perform an experiment using nanoparticle in stem cells in order to track the presence of this nanoparticle. In this case, in a model of motor neuron degeneration, we found very, very briefly, when we inject this nanoparticle, they are fluorescent in the tail vein, so systemically. We found that they arrive in particular in the liver, but remain for a short time. But on the contrary, when we when they were injected in the lateral ventricle, this is the control. This is white because this is plenty of water. And in this case, the MRI, it became black, the ventricle, because there is a decrease of signal related to the presence of iron oxide. And this is histology. We can see that here there are nanoparticles in red, but cells that can stay here and release some factor to protect this animal. So this is the three very, very brief example, but this is just to emphasize the philosophy of work that more or less remain the same. And just to conclude, I believe that the development of new technologies in biomedicine needs a careful characterization at preclinical level, and based on the study of behavior of carriers in terms of safety, stability, and accumulation, and other parameters before moving to the efficacy, and also the feature of tracers, contrast agents, fluorophore, and so on. We have to be sure that there is not biological elution, no bleaching, no toxicity, and so on. And another point important is, as I showed you before, the implementation with non-invasive instrument that can be somehow mimic what happens in a clinical study should be important in order to realize what is possible to see in patients. And finally, is another important point that quite often is neglected is that even when you start by proof of concept, you have to keep in mind that the adoption of robust statistical approaches when you choose and select the type of treatment are important, and also to comply with the ethical guidelines already at preclinical level. I just finished. I want to thank people working in my group and the audience, and many other collaborators, both in nanotech and nanomed field. Thanks.
Thank you. Yes. Thank you very much, Paolo. I have a lot of questions.
No, I close.
Now it's good. Okay, sorry. I have a lot of questions for all of you, but I think it's better to leave the floor to the space to some of the questions. Eric, do you have some?
Can you hear me? No.
Oh, you can come here.
Yeah. So I have a question. Thank you very much for the last presentation, for all the presentation. I have some questions starting from the last one. The first one is that in a model of inflammation in the liver that we saw, could you explain me better the model of disease that you produced?
Yeah, this is a model. Sorry. This model has been produced. It has been carried out by collaboration with a group in Frankfurt, and the model is it has been by the injection of a virus in mouse, and so there is an inflammation and fibrosis related to the presence of autoantibodies inside the mouse that they are developed, and then there's more or less like an hepatitis, and it progress and reach a plateau, and we can see some similarities than human inflammation or human autoimmune hepatitis.
Okay, so the administration of nanoparticles in that case was during the development of disease, or for prevention, or for the reversal of the disease?
We perform both, if I remember well, both type of treatment, because we tried before a treatment just at the beginning of symptoms, and then also, if I remember well, during the clinical progression. It's obvious that if, as you saw earlier, the results is better. But anyway, we found that there is the possibility to have a stronger and a faster response, and it was incredible because also the philosophy was to use nanoparticle and dexamethasone, not because dexamethasone doesn't work. Dexamethasone works because it's a steroid, but just to reduce the off-targeting, because dexamethasone is a lipophilic drug, so entering in all type of, more or less in almost all organs. And this was, and we were happy even if we managed the same results in terms of efficacy. But it was incredible because we found incredible, at least at the beginning for us. Then we think we have understood why, that in spite of the levels were at the beginning lower using nanoparticles, the results in terms of transaminases and inflammation, and the fibrosis was better than that we obtain with free drug. And we believe that is due to, I haven't enough time to show the results, but they've been published. We believe that the result was that nanoparticle enter inside macrophages. Filippo and Paolo already discussed about the macrophages, and very often they represent a limitation because they take an apple nanoparticle on drug. But in our case, they are they move for the amplification of disorder. So I believe because they're more subscribed to macrophages. So this was the main result in terms of efficacy.
Okay. The last question is, did you observe any zonation in terms of reduction of inflammation? I'm trying to contextualize better. So you observed that the inflammation in all the lobules were down in a homogeneous way, or was more periportal or pericentral?
Actually, we don't know because we perform experiments. You are perfectly right, and maybe we can try.
Because I think that the level of oxygen through the portal it would be nice to make like after the treatment the single cell analysis and understand
it will be important. You're right. You're perfectly right, and we have to perform other experiments. We perform experiments by RT-PCR, and we saw that there were some genes that were down-regulated in terms of. But you know that when you perform the RT-PCR, we haven't the microsurgery to take. But it could be an important point also because one of the, I think, advantages of nano delivery is that you can also try to direct.
I mean, I was thinking about that. You're trying to, when the nano delivery, you're trying to target nano objects, no? So it would be nice, like for proteomics, transcriptomics, have an overall idea of the pathways that are down-regulated, up-regulated, and after with single-cell analysis, understand which compartment, zonation, sub-cell analysis. So you know exactly which type of cells within the lobule is more interacting with the nanoparticle. That would be a nice.
You're perfectly right. We exclusively perform co-localization in terms of nanoparticle and the cells, but it could be maybe we can discuss. Thanks a lot.
Thank you.
Yes.
I would have another question for
Decuzzi.
Paolo.
Yeah.
Yes.
Yes, he's here. Okay.
Can you hear me? Paolo.
I am alive. I can hear you. I can see you. So.
Presentation. I really enjoyed your presentation.
Thank you.
Really nice. I have a question about the fibrotic experiment, the reduction of fibrosis with nanoparticles with the model CLL4. So the question is, did the nanoparticle was administered during the formation of fibrosis or after to the reversal of fibrosis? Second question, did the fibrosis was stage 2, 3, or 4, or did reach the cirrhosis?
Yeah, thank you, Eric. So you're referring to the methyl palmitate nanoparticles, and the reference to the fibrosis was because methyl palmitate is known to have also some anti-fibrotic activity, but we never tested it in that context. So it was just a reference, but again, we never tested that in the context of liver fibrosis. However, given the fact that I have seen also this morning, you are posing multiple questions that are related to hepatocentric diseases. I mean, if you have an interest in that, and if you have access to animal models, I'll be more than happy to start discussing how we can collaborate. So far, the methyl palmitate, and I think, but again, I flew through the slides. So the methyl palmitate has been using only for inducing a dormancy state, a transient dormancy state into the macrophages. We did not exploit the actual anti-inflammatory, anti-fibrotic direct activities, so the direct therapeutic activities of methyl palmitate. So again, if you have an animal model of liver fibrosis, where we can actually start looking into also the therapeutic efficacy per se of these particles, I'll be more than happy to look into that. Very happy.
Nice, for instance, to test some functionalized nanoparticles and explore some different type of fibrosis, like CLA, but also TAA and or portal hypertension driven fibrosis and both for prevention and for the reversal, especially for the reversal. Because I think that the problem is that the reversal. So when you look on the prevention, actually the anti-inflammatory effect is the one with any anti-inflammatory effect you can reduce the fibrosis when you prevent the fibrosis. The problem is that we need to reverse the fibrosis. With the CLL4, you know that when you stop to administrate CLL4, you reverse the fibrosis in mice and rats. So maybe with TAA model, the thioacetylamine model that is more stable could be interesting to test different nanoparticles. That's really, I think that it's a really interesting.
Eric, let's schedule a 30 minutes Teams or Zoom within the next few days, and let's talk more into the details. Again, as if you have access to the animal models, I can send you the particles early next week. It's easy.
Let's keep in touch.
All right, drop me an email. Drop me an email. Sure.
Okay. Thank you, Eric. If you have other questions? No. Okay. Okay, don't worry. So, thank you. Filippo, if you have a few minutes, I have a question for you. Yeah, if I didn't miss. Is there a microphone?
Maybe mine.
Okay. Okay, sorry. Yeah, I hope I didn't miss something that you said. You described different nanodrug, nanostructure for targeting specific cells for cord injury. And then at the end of the presentation, you say further perspective and that hopefully is to combine the effects of different nanoparticles in a single structure. Correct? So can you share with us how difficult is the path, or at which stage you are on this, the challenges?
A very challenging point, because in this kind of disease, we said we have different mechanisms that take place at also a different time. So at the end, of course, we need to combine them together, but allowing the proper efficacy at different steps. We tried sometimes when we were younger and without any kind of scientific guide to combine them just at the beginning, but it is something that is impossible and unfeasible, because you cannot understand what is wrong in on. So we decided to simplify the system. We have another parallel research line on stem cells that will seem to try to replace the damaged tissue, so with the injectable hydrogel that can carry the cells. So we hopefully we would like to combine hydrogels and nanoparticles that can be released at different steps in order to have a strong amelioration. We have also to understand to think that this kind of disease, even if it is extremely dramatic, it is different from the that the other colleagues also told about. So even an amelioration is fine. Probably we, like scientists, we look to the most challenging goal. So to have people that now it cannot walk, that can run and may compete at the Olympics game, but probably also a middle. So to have a good amelioration is would be a winning point for them for the quality of their life.
Thank you. Thank you.
Yes. I have to switch on my microphone. So, can you hear me? Okay. I have a curiosity about the results showed by Paolo. In terms of, you show, Paolo, if I'm not wrong, that there was in an experiment that the ratio between the level of nanoparticle in liver and lungs, the ratio lungs-liver, we can say, increases, right?
Yes.
So, but it is exclusively due to a decrease of level in liver, or there was also a increase of nanoparticle penetration in lungs? Sorry, I didn't understand this part.
Yeah, so this is the experiment where we...
I can't hear you.
Okay.
Can you hear me now?
Yeah.
All right, so this is the experiment where we actually use the methyl palmitate nanoparticles to somehow detour the rigid particles from accumulating into the liver. So in that experiment, we've been using rigid discoidal particles of about 2 microns. So these particles would end up into the liver as well as into the lungs. Dramatically. In fact, under control condition, meaning no methyl palmitate nanoparticles are injected, we saw a ratio between lungs to liver of 1.5 on average. When we pre-treat with the methyl palmitate, what happens is that we basically mostly hit the Kupffer cells. And so the ratio that under control conditions, so without methyl palmitate nanoparticles, was 1.5, now starts to grow. So 2 hours post-treatment, it was about 2 point something, and at 4 hours, which is the maximum, it was close to 4 times. So there was a reduction in the accumulation into the liver and an increase of accumulation into the lungs. This was also clear from the data that we collected with histology and also with the fluorescence by looking at the organs explanted and looking at the fluorescence associated with the particles. So there was a change in accumulation between liver and lungs.
Sorry, and just like curiosity about histology, have you seen if these particles remain in lungs, remain trapped inside the vessels, I don't know, the endothelia, or are able to penetrate inside, even if the lung parenchyma is not so plenty of cells, but they are able also to somehow interact with epithelial cells?
They were mostly either on the vasculature or perivascular. These are relatively big particles. These are a couple of microns, and they were basically seen in the lungs, mostly in the vessels, so sticking to the vessels or immediately perivascularly. They were not able, and you're right. I mean, if you look at the histology of the lungs, you start questioning where are the cells here, right? So you see spot of cells, right? So it's not super dense as it would be a tumor, but no, they were not traveling deep into the lung. But of course, the idea was that of demonstrating that we can control the accumulation in the liver versus lung by blocking the liver and accumulating more particles into the lungs.
Yeah.
Okay, thank you. We have a bit of time for other questions, for a discussion.
No, perhaps I can, because this was a comment. Paolo expanded it a lot, but Filippo mentioned it, right? When he said we're trying to simplify the system for reasons that are clear to everybody. I'm not going to discuss here this, but clearly he had in mind, the complexity of translating it into the clinic, right? And Paolo mentioned something that is extremely important because he said, one of the issues is that we cannot find CMOs and CDMOs that can actually manufacture either at the preclinical, like the CGMP-like or CGMP particles. And that is totally true. It has been limited a lot, the field of nanomedicine. One of the reasons why we have also that big valley of death with the skeletons at the bottom in a very graphic image that presented by Paolo is also because there are many nanotechnologies, nanomedicine that have been developed, but we are not nanotechnological or nanopharmaceutical technologies, right? To mimic the pharmaceutical technology, the terminology used by the pharmaceutical people. And that's what we are really, I think, missing right now. And I'm confident that within the next year, we will see more and more and more. We have seen the microfluidic, right, revolution with all the system produced by different companies in Europe as well as in Canada, in the US, where we can actually produce liters of lipid-based nanoparticles, polymer-based nanoparticles with a very well-defined size. But if you want to move away from that, there is still a desert, right? So we really need to invest more, in my opinion, into the nanopharmaceutical technology. So in really developing technological tools that would allow the industry to produce our tools, our, let's say, nanomedicines at a higher scale and under CGMP conditions. So that was the comment of Filippo that was, within all the signs that have been presented. It just mentioned it very briefly, but also the slide of Paolo are extremely important and really are the distance, they somehow signify the distance between industry and the academia in this case. We need to learn how to fabricate our stuff. And so going back to the skepticist, right, they always make the comparison with the pharma industry, with the small molecules. Small molecules have been synthesized for 100 years. We know everything, right, about that. We know how to scale up. We know how to do the GLP talks. We know how to do all the quality check. Nano is still very much to be discovered. And I think that there is need for real technologists in the field, and many, many, many engineers should join in.
Just to conclude, indeed, I completely agree with your wish when you said that we hope that after the approval of Moderna or Pfizer solid lipid nanoparticle vaccine, there will be also a cultural change in which also regulatory or maybe other phase will be easier. I don't know which is your idea. This is my a wish, but I'm not so sure. I hope yes, and it will be a very, very important point because, as Paolo said, very often people working in big pharma, where we can say very, very skeptical about also for economical point of view and so on.
Yeah. Yes. Well, as I said before, I had really a lot of questions, but we are running late, so maybe I will send an email to Paolo as well, Paolo Decuzzi. Paolo Bigini is very close to me, so I can reach him in a very easy way.
By voice.
Yes. By voice, by phone. So I really would like to thank you all the speakers of this afternoon. It was a amazing session for us, especially for people who are not so into the nanomedicine. We learn a lot. I hope the same for all the other attendees. And thank you, Paolo Bigini, Paolo Decuzzi, Filippo Rossi, and again, I would like to thank a lot Michele Diana, Stephane Cotin, Eric Felli, Martina and Andro. I hope you enjoyed this day. The aim was, as I say, to give food for thought and also to try to establish some connection. I hope we could achieve this goal. At least for me, it was a big pleasure to see many friends again, to meet them again, and hope we will see in person soon. So thank you for your time.
It was a really amazing and nice mixture, nice cocktail, and I really appreciate a lot. Thanks a lot.
Thanks. Thank you, Michele. So, all right, you are free. You are free. Have a good afternoon and keep in touch.
Thank you very much, everybody. We'll stay in touch. Bye-bye. Bye-bye.
Bye. Bye.